Now it is going. Zoom to YouTube. ಹೇಳ್ಕೊಳ್ಳಿ ಚೇರ್ ಹೇಳ್ಕೊಂಡು ಕೂತ್ಕೊಳ್ಳಿ ಒಂದು 10 ನಿಮಿಷ
Hello,
ಆರಾಮಾಗಿ.
Rudresh sir. How are you?
Fine, sir. How are you?
Yeah, fine.
So, good morning.
Yeah, yeah, kind of good evening for us. For you, good morning. We are still in 10th. You people will be in 11th.
Yeah, because here the problem is people are not ready to get into the mood of what we call as academics early in the morning.
We will
That is
have
the
lab
problem.
meeting at 8. For us, lab meeting is at 8.
Okay, but here
Morning 8
is
a.m.
things are different. And now here winter season, no, actually what is happening, the temperature is so kinetic energy is low. They won't get up early. That is one problem.
Yeah. Even in demands, it was like that for us. Like morning meetings are usually mornings.
No, Niman's condition is different, sir. Here, all these people, no, they are not from Nimans. That is a problem.
Yeah, I can
They take
understand.
their own time. That's the this one. What we call as the curiosity is not there with the people. That's the problem. They take everything lightly. Let it go. Let it happen like that. We'll see.
Okay. There are people are from Nimans also. Looks like some people maybe by name I can make, or maybe Nimans.
Yeah, people are joining.
Hi, Devraj.
Hi, Maitri. How are you?
I'm good.
How
I'm good.
Hi,
Hi,
Maitri. How are you?
I'm good. How are you doing?
Yeah, good.
Nice. Nice to await your talk.
Sure. Hope
Hello.
you guys will able to follow me and enjoy it.
Yeah, sure.
So
Hi,
who else from
Rudresh.
Nimans?
Yes. Srinivas Bharat.
Oh, okay.
ಮಾಡೋಣ. But still people are registering.
I think they can join in if you want me to start, or I can wait another.
Yeah. 5 more minutes we'll wait because participants are on 35 now. Yeah. Slowly people are joining. ಸರ್ ಎರಡಕ್ಕೂ ವಾಟ್ಸಪ್ಪಲ್ಲಿ ಬಿಟ್ಬಿಡಿ ಲಿಂಕ್. ಅದಕ್ಕೆ 2 ಬಯೋಕೆಮಿಸ್ಟ್ರಿ ಆ್ಯಂಡ್ ಇದಕ್ಕೆ ನಮ್ದು ಇದೆಯಾ ಬೇರೆ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ಗಳ್ದು ಏನಾದರೂ ಮಾಡ್ಕೊಂಡಿದ್ದೀವಾ? ಇಲ್ಲ
ಇವ್ರು ಬಿಟ್ಟಿದ್ದೆ ನಾನು ಟೀಚರ್ಸ್ ಬಿಟ್ಟಿದ್ದೇನೆ.
ಇದಕ್ಕೆ ಬಿಡಿ.
Okay, Devraj, we'll get started. You are on mute. You are on your speaker. You are to unmute.
Yeah, 5 more minutes.
Okay.
Getting delayed.
Oh, no. I thought I will just start introduction. So at least people can join in as we go on.
ನಾನೇ ಬೇಕಂತ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿದೆ. ಎಲ್ಲೋಯ್ತದೆ? ಹ್ಮ್. Yeah, I think we'll.
Yeah. Okay, Devraj, I'll start introducing myself and.
Yeah. No. We will wait. We'll formally welcome you. So I have given option for sharing as well. You can share.
Okay. Okay.
So good morning, all of you. I think some are waiting. I'll just admit them.
You can see my slide?
Yeah. I can see.
Okay.
ಇಲ್ಲಿ ಆಫ್ ಮಾಡೋಣ ನಾವು, ಅಲ್ಲಿ ಎಂಟಾಗುತ್ತಾ? ನೋಡೋಣ, ಎಕೋ ಬರಲ್ಲ ಅಲ್ಲಿಗೆ. ಯೂಟ್ಯೂಬಲ್ಲಿ ನೋಡಬೇಕಿತ್ತು ಎಕೋ ಬರುತ್ತಾ ಅಂತ. ನೀವು ಯೂಟ್ಯೂಬಲ್ಲಿ ಚೆಕ್ ಮಾಡಿ. ಇಲ್ಲ ಬೇಡ, ಅವ್ರಿಗೆ ಹೇಳ್ಬಿಡಿ. ಹೊರಗಡೆ ಅದಾರ ಪ್ರವೀಣ್? ಓಕೆ, ವಿಲ್ ಸ್ಟಾರ್ಟ್ ಆ್ಯಂಡ್ ದೆನ್ ವಿಲ್ ಚೆಕ್ ಇಟ್. Good morning to all of you. Welcome back to the international webinar on recent trends and challenges in immunology. So, which is being organized by the Department of Biochemistry, Karnataka University, Dharwad. So, I am. I am audible. Good morning to all of you. Welcome back to the international webinar Yeah, that's fine. I think. Yeah, today here with us, Dr. Ram Kumar. He is going to talk about epilepsy, understanding the mechanism of Sudeep, and he is a good friend of mine. And so I ask, formally welcome, Dr. Ram Kumar to this session. So he is going, our faculty member, Dr. Vishwanath Chachadi, who is an organizer, coordinator of this event. So he is going to introduce you formally to for the benefit of all the participants. Okay, I hand over to this, to Dr. Vishwanath Chachadi.
Yeah, very good morning all. Myself, Dr. Vishwanath Chachadi. I'm an assistant professor at the Department of Biochemistry, Karnataka University, Dharwad. Once again, I welcome you all again for this seminar on understanding the mechanism of SUDEP, that is sudden unexpected death in epilepsy. It's my pleasure that I have been asked to introduce today's speaker, that is Dr. Ram Kumar Kurba, and who is going to deliver a talk on epilepsy, understanding the mechanism of SADAP. So Dr. Ram Kumar, he is an assistant professor, research scientist at the Department of Neurosurgery, University of Iowa Hospital and Clinics, Iowa, USA. And earlier, he did his undergraduate studies in biology and chemistry and masters in biochemistry. Later, he completed Master of Philosophy and PhD in Neurophysiology at the Department of Neurophysiology, National Institute of Mental Health and Neuroscience, that's NIMHANS, Bangalore, India. For his PhD dissertation, he investigated the effect of brain stimulation, reward, and exposure to environmental enrichment on chronic stress-induced structural, chemical, and functional deficit. For his postdoctoral research, Dr. Kuba moved to Duke University Medical Center, USA, where he worked on the role of hippocampal neural stem cells as potential therapeutic strategy in preventing seizures and epilepsy development. Later, he moved to Texas A&M University, where he worked on the acute toxicity effect of nerve agent and the role of neurosteroid as therapeutic agents to counter nerve agent-induced seizures and neurodegeneration. Currently, Dr. Kuba is working on sudden unexpected death in epilepsy mechanism at Department of Neurosurgery, University of Iowa Hospitals and Clinics, Iowa City. He has authored 12 research papers, 3 review articles, and a book chapter, and is also a reviewer for many scientific journals. He has taught scientific techniques and mentored many undergrad and medical students in summer research projects. He is also adjunct faculty at Kickford Community College at Iowa City, where he teaches anatomy and physiology. With this brief introduction, now I would like to request Dr. Ram Kumar to take over, and you can proceed to the talk, sir. Thank you so much.
Yeah, thank you for your introduction. And good morning to everybody. So today I'll be presenting my research work. It's on epilepsy, mainly looking at the mechanisms of what is known as sudden unexpected death in epilepsy. It's called SUDEP. You might have heard of it. Recently, there was a Hollywood actor, young actor who died of SUDEP. So this has been there. Sometimes the press picks it up and we come to know, okay, even some famous personalities too have this happening. So before we go into the talk further, we should know like what is epilepsy, or what is the difference between epilepsy and seizures. So epilepsy is a disease where two unprovoked seizures can occur, and the difference in between them could be 24 hours. That's the official definition now from International League Against Epilepsy. That's they defined it in 2014. So then the question is, what is a seizure? So a seizure can be a transient occurrence which causes signs and symptoms because of abnormal excessive and synchronous neuronal activity or abnormal firing activity in the brain. So this may cause unconsciousness, involuntary movements, so those kind of things. So in common terms in India, we call it fits, okay? So why we should study epilepsy? Why is it important? Because it is known now around 70 million people around the world have it. And in India, there is a recent study which show that the prevalence is like 1%. So you can imagine, so there are around 12 to 13 million people who have epilepsy in India. So when we come to here to US, it's around 3.4 million. And what is worse is like of these people with epilepsy, in them around 30 to 40% of these people, they do not respond to any treatments. That means like their seizures are not controlled by any drugs, even though there are drugs available, but still their seizures will not go away. So one thing is the seizures are a huge burden on the person himself, because these are unpredictable. And if the seizure occurs when a person is driving or on a road, an accident can happen. And also, it is also a huge burden for the family, because sometimes people with epilepsy, they can't do many things on their own. So somebody needs to take care of them. So it's also a huge economic burden. So what are the causes for epilepsy? So there are multiple causes, many causes. It's difficult to pinpoint which is the one, but the causes can be broadly classified into these four groups, like the causes that can happen during infancy and childhood, the causes that can happen during childhood and adolescence, and during adolescence and adulthood, and in the older adults. So during infancy and childhood, usually it's most of the time it is prenatal or some problem during the pregnancy or during the birth that can result in brain injury, can sometimes lead to epilepsy. And sometimes there can be some genetic causes, like some inborn errors of metabolism in the child. They later, after the birth, can cause epilepsy. And of course, there is the congenital malformation, where the development of brain is not proper. So that can also lead to epilepsy. So coming to childhood and adolescence, here again, a genetic component can play a role. And usually during adolescence, there is a lot of infections happening, sometimes CNS infections, that is central nervous system, or the brain infection can lead to epilepsy, or trauma, that's any accident or injury to the brain. And even that's the same case here with the adults too. Like now, as the person move into young adulthood, because the person is very active, he can undergo any time any head trauma or head injury, like accidents, sports injuries, those things. And due to societal pressure and peer pressure, they may also involve in intoxication, like drugs, alcohol, and those things. And when they go into withdrawal, they can have seizures. And when it comes to older adults, usually it is stroke. Sometimes it can be brain tumor, or sometimes it can be neurodegenerative diseases like Alzheimer's. So most of the neurodegenerative diseases can also cause seizures. So when we look at, so when we look at what is happening in these patients or how the epilepsy happens. So initially there may be a brief seizure event because of a drug or injury, or it can lead to what is known as status epilepticus. So this is a condition where the seizures don't stop. They can last many minutes continuously and the patient is unconscious. So usually if the patient undergoes this condition, the patient is taken to the emergency. Or repeated seizures happening over and over. So this kind of seizures, what they do is they cause some injury in the brain. That is called initial injury. So in these patients, sometimes they will have seizures and then nothing happens for weeks, months, or years. So that is called a latent period where they don't get any seizures. But after a few months or few years, suddenly they show up starting having seizures again. So that is when we call the person is having epilepsy, because now the seizure is happening spontaneously without any provocation. So from the initial seizure that's caused by injury or a drug withdrawal or febrile seizures in children sometimes, to this phase of chronic epilepsy, there is this huge latent period, which may be months to years or sometimes weeks. So what is happening during this phase? So we discussed in one of our reviews based on the earlier studies, like, so whenever there is a seizure, there is a lot of release of glutamate from the neurons. So that leads to what is known as excitotoxicity that leads to apoptosis or the programmed cell death. It also leads to inflammation and glial reaction. It also leads to a lot of modification of the synaptic proteins. The synapse, that's like a space between two neurons where the signal is transmitted between neurons. And there is also transcription changes of the genes with regards to the structural proteins and the signaling proteins. So that is the initial phase. And during latent period, studies have shown there can be a lot of plasticity happening. That means there are a lot of structural changes happening. There is the reorganization of neuronal connections and also the firing pattern or the synchronized activity. So once the person starts showing the epilepsy, what the animal models have shown was usually now the seizure can be picked up with the EEG. You can see it as an electrographic seizure. You can see there are some degeneration or atrophy happening in some of the brain regions and some abnormal sprouting. Here in this case, mainly hippocampus, a structure that's involved in learning and memory. That is situated in the temporal lobe of the brain. That is at the just besides the ear. Okay. So how the seizures are detected? So usually seizures are detected with the electrodes placed on the scalp or on the head, and also with the video. So it is called video EEG. Okay, so you can see here when the person has these abnormal firing activity. Okay, so those are considered like electrographic seizures or the seizure activity. So here seizure activity, first it can show just on the EEG, the person may not show any symptoms. But when the person shows the symptoms, it is called generalized tonic-clonic activity, where the person cannot maintain balance, he may collapse, he may get unconscious, and involuntary limb movements. So all those things can be seen in the person having a seizure. So coming to the treatment of epilepsy. So even though we have like around 20 anti-epileptic drugs that are approved by FDA, so not all of them are effective. Because as I mentioned earlier, 30 to 40% of these patients they have what is known as resistant epilepsy or medically intractable epilepsy, where even with one drug or combination of drugs, the seizures are not managed or reduced in these patients. So these patients are called medically intractable epileptic patients. So these patients are usually the candidates, those undergo what is known as epilepsy surgery. So here it is a kind of a calculation between cost and risk ratio. Because the seizures are so bad in this person, he cannot lead any normal life, he cannot do any things on his own. So people opt to go for epilepsy surgery. So essentially what the neurosurgeons do here is they map out with EEG where the seizures happen, that is try to localize the seizure focus, and then do the surgery and essentially take that brain part out. So it helps some of these patients, but again, there is that risk, the patient may lose some of the functions. So it is always a benefit risk ratio. So here, based, as I mentioned, so based how the patient is showing seizures, where the seizures are originating. So just to give a idea here, this is the human brain. When you're seeing from the side, this is the temporal lobe, this is the cerebral cortex, this is cerebellum. So cerebral cortex is divided into different lobes like frontal lobe, parietal lobe, and temporal lobe. So usually when they record the EEG, sometimes just the EEG from the scalp will not be very clear in the resolution of the seizure focus. So what they do is what is known as intracranial EEG. So essentially they open the skull, they open the meninges, and then they place these electrodes directly on the surface of the brain. And then the patient is made awake. So he has all these leads, and the patient is recorded 24 hours for 2 weeks to see how, and what they do during this phase is they reduce the amount of drugs. So what happens? The seizures may happen. So since the patient is in the hospital, they're taking care that seizures don't get really bad, because they want this to do so that they can see where is the seizure focus, from which area of the brain the seizure starts. So they want to localize that and then do surgery and take it out. So this is the temporal lobe from the side, okay, the frontal and the parietal. So here, this patient is placed, the electrode leads all over the brain almost. Okay, so these studies here are done here at the University of Iowa hospitals. So where there is a big research group where I'm part of it, we study what is known as SUDEP, sudden unexpected death in epilepsy, in these epileptic patients who are resistant to drugs. So what is SUDEP? Because usually why these patients are scared of this condition is, excuse me, these patients seem to be healthy, their seizures are controlled sometimes, but suddenly they are found, one fine morning, they're found dead in the bed. So these patients, when they check, then the doctors check with their relatives, they don't see any seizure or anything happening. And there is also no evidence of a seizure. Excuse me. So with regard to the incidence, there's almost 50% of SUDEP, chances of SUDEP happening in these patients who are resistant to drugs. So, and most of these patients are also young. Excuse me. And this SUDEP, it causes a loss of life when they're young. So it is only second to stroke with regard to the potential years lost. So why does this happen, SUDEP? And what may be the causes? So it is known, like when in some of the patients, when seizures happen, there can occur what is known as loss of breathing, or also called as apnea, but it's also called central apnea. That means the patient stops breathing for a brief time, for some time. So what this causes is, it causes decrease in the oxygen level in the blood, and this may be the major cause. That's what we were thinking, but we have to show it. So based on the mortality reports of these patients, and based on the literature, like when the patient is having seizure, there may be respiratory dysfunction. And when these patients are found dead, they are found in prone position, that is face down in the bed, and often in sleep. So then what Dr. Dilovi, he is the neurosurgeon I'm working with. So what he and others showed was, like, these patients, when we implant electrodes and started studying, because this surgery, when they do it and implant the electrodes, it gives a very good opportunity or a window of opportunity to study during seizures what happens to these patients, because these patients are inpatient in the hospital. So we used all this respiratory monitoring unit, like where there is nasal airflow pressure, because different parameters like oral thermistor, abdominal belt, chest belts. So we can capture what is happening when the patient is having a seizure. So this is how typically it looks when patient does not breathe. You can see here in all these parameters, the nasal flow, the abdominal movement, the chest movement, suddenly they become flat from this wavy pattern. So what Brian found was, like, when these patients start having seizures, and when the seizures spread from cortex to left amygdala, that is a temporal lobe structure that's very near to the hippocampus. The temporal lobe, I showed you that. So it is in that region. So what happens is, suddenly the patient is breathing when he starts having seizures, but at specific time point when the seizures spread through subcortical structures like temporal lobe structures like amygdala, that's involved in emotional learning, what happens is the patient stops breathing. Okay, so, and this breathing is not just few seconds, okay? So it lasts for almost a minute. So, and then once the seizure stops, then he will start recovering he will start coming to normal breathing pattern. Okay. Yeah. So essentially that's what we are seeing. Essentially, to support that, when we record blood oxygen levels, there is a rapid fall in the oxygen level. Okay. So this was kind of a landmark paper, because up till then, the usual kind of idea in the field was, it is seizure happening, and then maybe something happens to the heart, is a problem with the cardiovascular system, and then the patient dies. But this study showed very clearly that it is first the respiration that is shut down. Then once the oxygen level falls, that can lead to cardiac problems, and finally cardiac arrest. And then the patient may die. So just to see, okay, this is happening during seizure. So since we have this opportunity, the patient is in the hospital and is monitored and taken care. So Dr. Brian Dilovi, he went ahead and wanted to see when the patient is not having seizure, what happens to this patient when the amygdala is stimulated. So when this patient's amygdala is stimulated, same thing is seen. The patient stops breathing. Okay, so again, same way, the blood oxygen level falls. But what's important is here, the patient is just sitting in his bed and stimulated. He's not told anything, okay? And doctors ask him, like, when he has stopped breathing, and do you feel any change? So usually what happens in a normal person, whenever you try to hold your breath, you cannot hold breath for a long time. That urge to breathe is there. So that is impaired. It's called dyspnea. So, and here the stimulation is almost for 40 seconds. And these patients, when you ask them to hold the breath, they cannot hold it that long. And very dangerous thing is this patient is not aware that he is not breathing. So stimulation of amygdala is changing something in this consciousness or some perception, change in perception, where his system is not recognizing that he is not breathing and he needs to breathe. So this gave a very good insight why these patients are found suddenly dead. Because imagine a seizure happens in the middle of the night, the patient is sleeping, there's nobody observing the patient, and if something like this happens where he stops breathing, and if by chance, if the patient is on face down, so virtually it is like the patient stops breathing and then it can lead to choking and subsequently heart failure. So this was kind of like a major landmark in this field where directly our group showed that in patients, this is happening. So, yep. So the most interesting thing was these patients were not aware that they were not breathing. So it just shows how important it is to be conscious about breathing. So something changes in their brain because of epilepsy. So these patients, when the seizure spread to temporal lobe structures, they are not breathing. So we had this hypothesis, like when there is a seizure that spreads to amygdala. So this amygdala has connections to brainstem regions, which control the arousal and awareness, and also breathing. Like brainstem also controls, as you know, the breathing and the cardiac regulation. So when these patients' brainstem is affected because of seizures, that is in turn spread to amygdala and controlled by amygdala. So finally is resulting in loss of oxygen in the blood, I mean, decrease in blood levels of the oxygen, and then finally leading to the cardiac arrhythmias. That is, the heart may starting beating faster because it is a stress to it, and it may also lead to cardiac arrest, and then lead to the death. So just to summarize the human studies, so what we found was seizure spread to the amygdala, caused loss of breathing. And the more worrying thing is the patient is unaware, and that can lead to SUDEP. And just to as a proof of principle, when the seizure is not happening, just with a normal amygdala stimulation also causes loss of breathing. So it becomes very important identifying patients where seizure spread to amygdala and that may interfere with breathing and try to develop preventive strategies. So this is the work our group is doing, and I am trying to work in a reverse translational way where I'm trying to develop an animal model. Because when we want to do mechanistic studies, animal model is a very good thing, because in patients, you can only do that much because there are a lot of ethical concerns. So here I try to develop a mouse model of epilepsy. So just to give you an idea, we can induce epilepsy in rats and mice with chemicals known as chemoconvergents. Here is an example. When we inject pilocarpine, after a half an hour or so, these rats start showing the seizures. So these seizures are graded according to Racine scale. So here just what you saw was a level 4 seizure. It is a little higher intensity seizure, where you can see the rat is sitting and it's moving its both forelimbs. And we'll have what is known as the highest level, that is the level 5. So it does that forelimb clonus, but it also starts losing the hind limb balance and it starts falling down. Okay, so this is called a Racine scale from 1 to 5. 5 is the highest level of seizures. So with this setup, what we did was like, I essentially set up the same kind of setup that as used in humans to record the breathing from these animals. So I implanted these mouse with the electrode and then stimulated them with the current, and then the breathing is recorded and amplified. And excuse me, so you can see here how the mouse is normally breathing. When I stimulate amygdala, I can mimic exactly what we saw in humans. So it shows that these structures act very similarly, or they have very similar connections, and they show the same effect of electrical stimulation. The mouse stops breathing as long as the stimulation happens. And then it has this huge gasps, and then the respiration or the breathing resumes. So I implanted the electrodes here, okay, very near to the amygdala. And this is how, when I cut the brains, I can see the location of the electrode tips. So this is not just one time, okay, you get it, or it is very, very consistent in these animals. So you can see here on the left central amygdala, when I stimulate and I do 10 trials, 10 times, in humans we did 3 or 4 times, because there is a danger the patient may develop seizure, because we have to be very, very extra careful. But in these mice, in the normal mouse, when I stimulate under anesthetic condition, 10 trials, each time I stimulate, I can see the same response. The mouse stops breathing. So it's just not one side, it's even the opposite side. So we have bilateral amygdala on both sides of the brain. So both are responding to the stimulation, when you stimulate them, they induce cessation of breathing or central apnea. So this gave us a very important model or a tool to study how we can go ahead with this study. So right now we are building up this as a model and publishing a paper saying that we have this observation from humans, very similar observations we can see in the animals. So next we'll be doing it in the epileptic animals. So that's the plan. So essentially what our group has studied and shown is, which make a very important shift in the field of SUDEP. That is, the seizures, when they spread to the amygdala or temporal lobe structures, they shut down breathing. And then the cardiac abnormalities happen and the patient is at risk of death. So that was our major contribution, how the structure like amygdala is so important because it has connections to the brainstem structures that contains awareness and also breathing and heart rate. So I will stop here. I would like to acknowledge my PIs, Brian Delovey and John Wemy, and my lab members, Gail, Madison, and also the funding from NIH. Yeah, I think just as a side thing, so do you know what to do when you see a person having a seizure? What is the first aid to be given to the person? So these are the 6 things they suggest. The Epilepsy Foundation: stay with the person, time the seizure, that is check how long the person is having the seizure, keep the person safe, move away all the harmful objects. And if the person is lying down, make the person to be on the side. Don't block the airway. Don't put anything in the mouth. And then do not restrain. If the person is having seizures and his arms or legs are moving, do not restrain. And after a while, the person may wake up. So stay with the person. Usually when the person wakes up, he may be confused or disoriented. So you have to help him with that. But it becomes more important to call medical help when the seizures last more than 5 minutes. And seizures are coming one after the other. The person is having problem breathing. And also it is very dangerous when the person is in the water or if he's driving, if he's having seizures. So it's very important to monitor these patients. Yeah, so essentially the SUDEP is these associations, which are mainly patients, caregivers and all. So they spend a lot of time and come up with these kind of ads. So these are some of the risk factors for SUDEP. Means the seizure should be well controlled. The patient should get enough sleep and no alcohol. If at all alcohol, very, very less. Yeah, I think I'll stop here and I'll take the questions. Yeah, thank you. Okay, anybody has a question? Any doubts or clarifications? Okay, there is a question on by Dr. Manohar Patter. Do the epigenetics helpful in managing the epileptic effects on affected individuals? I am not clear what you mean by epigenetics. You want to ask that question or talk about it? Another question from same person is like, is there any studies on use of acupuncture and treatment of epilepsy? And is epilepsy acupuncture alternative therapy by directing nerve stimulation? I'm not aware of acupuncture as a treatment for epilepsy, but there are some experimental or investigational studies where in some cases when the seizures are not controlled in these patients, they use what is known as vagus nerve stimulation. So they implant a stimulator. So that will be having the leads on the vagus nerve. And it will be like a kind of a loop system. Whenever there's seizure going to happen or seizures happen, the stimulation starts automatically. So it is inbuilt system. I've heard about those kind of things, but acupuncture, I'm not aware of. Okay, another question by Dr. Pramod. How do we initially identify epileptic attack in infants? Is it congenital? In infants, it may be congenital, but sometimes it can also be febrile seizures. It depends, like if the infant is having a high fever, that can also cause seizures. And without, if it is not a febrile seizure, it can be a congenital thing too. How do we identify it? That would be difficult because if you doubt the infant is having epilepsy, it's better to check with the neurologist. So they will do the EEG and find out. Behaviorally, it will be difficult to say because the infant cannot talk and say what he or she is feeling. Only one thing could be sometimes these infants, they must, if they are eating or sucking milk, they can stop the activity and start staring. That is known to happen sometimes, but it's very difficult to make out in infants. So better to check with the neurologist. Okay, the next question. Okay, Shahid Hussain Khan, epilepsy is totally preventable or not? What do you mean totally preventable? Because as I mentioned, the causes are not very clear. You never know why the person got epilepsy. It may be some infection, it may be some injury some long time back. As I told you, like during injury itself, sometimes the seizures may happen, but the person may not know. So then later they may develop epilepsy. So preventable, that is difficult. We still don't have any drugs that can prevent epilepsy as such. So all these drugs I mentioned, the 20 drugs that are approved by FDA, these are mainly treating the symptoms, stopping the seizures. They're not reversing the disease. Okay. Yeah, we're still not there. Okay, Dr. Dayanidhi, is the people suffering with epilepsy have low IQ? It depends. It depends on the seizures intensity and the number of seizures. Like some people will have maybe like 1 or 2 seizures per day. Those people could still manage many things on their own, but some people will have a lot of seizures. So because seizures also affect brain development and learning and memory, and sometimes they also lead to depression. So it is possible they can have low IQ depending on how severe the disease is. Okay, then again, Dr. Manohar Patel, change of living environment of patients. Yeah, this would come more about managing the epilepsy of these patients. First thing to take care is the seizures are really controlled well with medication, or if possible, if seizures, uncontrollable seizures, maybe with surgery. Yeah, if it is a good environment after that, then it is obvious it can be helpful for the patient. Other question is continuous medication. Yes, continuous medication is very, very important, because some of these patients, even if they forget taking drug for a few days, the seizures may come back again. So that's why the very strict regimen of taking medication. Okay, Dr. Rashmi. Okay, the condition of epilepsy can be identified before the childbirth. Yes. Sometimes I'm not sure how much literature it is known, but I know one of the patient was lady was pregnant, and she could feel when the child in her womb had seizures. So it can happen during the pregnancy to the child. Okay, and is the epilepsies and age and gender biased? I'm not sure about that, but usually epilepsy is known to be more prevalent in men, because one is trauma, injury, accidents, because those activities are highly performed by men. Women may get, but it depends. But usually we see a lot of men with epilepsy. Okay, complex partial seizures. So there are a lot of seizures. Epilepsy is not just one disease. Like when they say epilepsy, this person can have temporal lobe epilepsy, partial epilepsy. So these are different terms used. Sometimes complex partial seizures, the person may not have overt limb movements, but he may pause when there is a seizure. But when we record with EEG, we can make out he is having a seizure. So there are different names for different kinds of seizures. Okay. Again, Dr. Manohar Patel, does meditation play any role to manage epilepsy? I would say medication is the first medicine. That is the first priority. Because if person is having epilepsy, medicine has to be taken. These things come later. Meditation, good conditions, living conditions, and all that. Maybe they may reduce the frequency of seizures. Excuse me. Excuse me. Okay, SN Thanu. If someone had epilepsy in childhood and treated, is there any chance of it recurring? Excuse me. Yes, there are chances. It depends how they were treated and if the person had been told to take medication, or what can happen is sometimes now it is being studied in animal models too. Usually when the person gets epilepsy and they may not develop later any seizures. I mean, once the person has some seizures and later they may not develop any epilepsy. So since they already had seizure once, and later if they get into some kind of problem with like a brain injury, excuse me. So it's called second insult. So the first insult has already made the brain more susceptible. So the person may develop epilepsy. It depends. Like if the child had epilepsy and treated and nothing happened, but later if the child gets any brain infection or any fall or any brain injury, then the chances of getting seizures are more. How long would it take to treat seizures? It depends. That's why I told you, like 1/3 of patients is very difficult to treat seizures because they're not controlled by any drug, current available drug. So it depends. Like sometimes for some people, it is lifelong medication. They have to take seizure medications to control seizures. Other question, can we have seizures but not epilepsy? It depends. Like if it's just happening once or twice, and then it doesn't happen, the seizure doesn't happen, then no, it's not epilepsy. That's the case. Like sometimes when febrile seizures, when the kid has a high fever, the seizures happen, but later that kid may not develop any epilepsy. So such a thing can happen. Okay,
good morning,
next
sir.
question. Excuse me?
Good morning, sir. This is Rashmi.
Yeah.
I had asked a question like, will the epilepsy can be identified before the childbirth? Because like one of my nephew was born with this. Soon after 3 days of the birth, he got convulsions, and the oxygen supply to the brain was seized, and the left part of the brain was, it was created vacuum, and he was not having the motions, like locomotive organs were damaged actually. So it was because the doctor told before the birth it could be identified, but you people didn't had the treatment for that. So
Yes.
even after the birth, can it be treated so that it can be reduced and the brain damage would be reduced?
Yeah, as I mentioned earlier, so even during pregnancy, if the unborn child has epilepsy, it can be detected. So the mother, they say, can feel it, but I'm not sure how. But any something not normal, maybe that should have been checked. That's one thing. But as you mentioned, I don't know about the details, but if the child is having already some brain impairments or brain structural impairments, I think that may be a congenital abnormality. I'm not sure. So that will be very difficult to treat.
Okay, but to mention that the baby was born normal. It wasn't having any problem when it was when it was born. So soon after the convulsions, there was the problem started after the 3 days of the birth. So till then the baby was normal. It was normal, feeding was there, crying was there, everything was there, sir. So that is what I wanted to know.
Yeah. So did the doctors recognize or identify what was the cause of the seizures?
They told it was a sudden convulsions which happened, and it was not identified on spot. It took around 2 hours for them for the doctor to visit, because it was in the mid of the night which happened.
Oh,
And it
so
took
these
around,
seizures were happening continuously?
yes, every half an hour once it happened, and none of the doctors were there to attend, and it took around 3 hours, more than 3 hours for them to attend. So because of the delay in treatment.
Yeah, because maybe if they had intervened earlier, it would have been better. Yeah, that's what, as I mentioned, like if the seizures happen for longer time and multiple seizures, so that is not good. It's really bad for the brain.
Okay, so within how much duration it can be treated, sir? A very short time of how much?
No, that is difficult to say, because it's not just one seizure. After the seizure, as I mentioned, studies have shown like a lot of changes are happening in the brain. So it is as soon as possible to go to the doctor. That should be that should be the main purpose or goal.
Okay, sir. Thank you so much.
Yeah, thank you. Other question from Dina. Can epilepsy be treated by deep brain stimulation? It is still not a real treatment, but research has been going on now. There are some studies which came out, I think, recently. They're trying for some of the uncontrollable seizures. They were thinking maybe deep brain stimulation can work. Yeah, it is still not a common treatment. It is still in experimental phase. Yeah, I think I checked all the questions. So anybody has any further doubts or clarifications needed?
Is it a genetic one?
Which one?
Epilepsy?
No, causes, there are many. So sometimes it can be genetic.
Okay.
Means sometimes some genetic change in the channels like sodium channel, calcium channel, potassium channels, or it can be some genetic mutation that causes abnormal brain development. So many factors. It is
Now,
unless the
are there
patient is
any
treated.
kind of genetic markers to be used for a molecular diagnosis?
I'm not aware of any molecular markers for epilepsy as such.
Okay.
What they do is sometimes these epileptic patients, sometimes they will have some genetic mutations, single gene mutations. So those can be detected where they will do the DNA analysis and then they make out, okay, what is the underlying cause. So that kind of analysis can be done afterward, but not like a marker before you can diagnose who is having, who is not going to have. That is difficult. That is not, still I'm not aware of that.
So is this kind of a thing, is a what is congenital marriages?
Yes,
Yeah,
in
such
India,
kind of a
that's
thing.
why
Yeah,
that's maybe one of the reason very high incidence of epilepsy.
epilepsy. Yeah.
Yeah, because if there are mutations, congenital, yeah, they are called consanguineous marriages. Yeah, marriage among relatives. So sometimes, if the bad mutations come together, they can cause seizures. Yeah, it's not only seizures. Sometimes it's also, some of the studies have shown there are behavioral impairments and those things in these people. Okay, if don't have any questions or any doubts, shall we stop here now, Devraj?
I think no more questions now from the audience. I think an exhaustive questions you have already taken up.
Yeah, most of the questions I answered. Yeah.
I hope everybody would have gotten help from the thing. Okay, I think we must thank on behalf of the Department of Biochemistry in Karnataka University for having done a wonderful talk, and also the two coming at late hours. Probably you would have gone for a sleep, right?
Yeah, time to.
Thank you so much, Ram Kumar.
Yeah, thank you, Devraj.
So,
Thank
yeah,
you for giving me the opportunity to share my research work
most welcome.
with your students. Yeah, thank you.
How metal toxicity induces seizure? There is one more question. I think the back end.
Oh, okay.
Is it?
Yeah, toxicity is also one of the factors. Yes.
Okay, I think
Toxicity can happen. That's why you see these people like people who metal toxicity it can induce, but not. It depends on the dose, how much and what when.
Okay.
And with regard to toxicity, I was mentioning like you see when these people, especially in India, when farmers commit suicide with all these organophosphates that are insecticides. So they are very toxic. So they cause toxic induced seizures.
Okay. So I think
what is that? Any information on nitrate in blood? Nitrate in blood regarding what aspect?
no.
I'm not aware of nitrate and seizures.
Toxicity, what he's talking about, probably, yeah. I think Dr. Udresh is asking, yeah.
Yeah. He's mentioning nitrate in blood induced by seizure.
Yeah.
See, seizures cause a lot of release of chemicals because the whole brain is firing, right? It is very abnormal firing. So we'll have a release. If you see nitrate, not on nitrate, when you see the blood profile or the CSF profile, you will have a lot of neurotransmitters, a lot of factors released. So they can't be a marker. So yeah.
Okay, thank you, Dr. Amkumar, for your wonderful presentation.
Thank you, Devaraj. Yeah.
So I think for all participants, I would have a announcement. So we'll be joining for another lecture. Okay. So by Dr. Manjunath, he's going to talk about immune responses to viral infection by 10:30, right? Yeah, we have at schedule at 10:30. So we will have the same link, and in fact, okay, we will not disconnect you guys. You can take a break and then you come back and join us, after a while. Okay, so maybe 10:15 or something. You can 10 minutes before you can join us. 10:30 by sharp. Okay, we will be starting, our next session. Thank you.
Okay, thank you all.
Thank you.
Good day. Have a good day. Yeah.
Yeah.
Bye.
Thank you. Thank you, Ramkumar, once again.
Yeah. Thank you.
Mhm. ಆನ್ಲೈನ್ ಬರುತ್ತೀರಾ ಬಂದ್ರೆ ನೋಡ್ಬೋದು ಈಗ ಬಂದು ಇಸ್ ಇಟ್ ಓಕೆ ಅಂತ ಪ್ರಿಂಟ್ ಕೊಡ್ತೀರಾ yes sir we will have to send this link to first again ಮಂಜುನಾಥ್ಗೆ ನಾನು ಕಳಿಸಿಲ್ಲ ನಿಮಗೆ ಈ ಲಿಂಕ್ ಅಲ್ಲ ಕಾಪಿ ಉದ್ದೇಶವನ್ನು ಅಡ್ಮಿಟ್ ಮಾಡಬೇಕು ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಡ್ತೀನಿ ನಾವು ಮಾತಾಡೋದೆಲ್ಲ ಬುಕ್
ಈಗ ನೋಡಿ
ಬನ್ರಿ ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ರಿ.
ಒಂದು ನಿಮಿಷ ಮೊಬೈಲಲ್ಲಿ ಇದ್ದೀನಿ, ಒಂದು ನಿಮಿಷ.
ಇಂಟರ್ನ್ಯಾಷನಲ್ ವೆಬಿನಾರ್ ಅಂತೀರ, ನೀವೇನು ಒಂದು ಸ್ಕ್ರೀನ್ ಹಾಕೊಂಡಿಲ್ಲ, ಅದಿಲ್ಲ ಏನಿಲ್ಲ ಅಂತಾನೆ ಬಾಬಾ. ಅದೆಲ್ಲ ಸ್ವಿಚ್ ಬೋರ್ಡು, ಕೋಲು, ಮೂಟೆ, ಅದು ಡೋರ್ ಎಲ್ಲ ಕಾಣ್ತಾ ಇದೆ. ಹಿಂಗೆ ಎಸ್ ಪ್ಲೀಸ್ ಡಾಕ್ಟರ್ ರುದ್ರೇಶ್ ಬನ್ನಿ. ವಿ ಹ್ಯಾವ್ ಮೆನಿ ಅದರ್ ಥಿಂಗ್ಸ್ ಆಲ್ಸೋ. ನೋಡಿ ಇಲ್ಲಿ ವಿತ್ ಮಾಸ್ಕ್. PP kit, even PP kit we have.
Many people are live, sir.
ಯಾರು ಅದು PPP ಕಿಟ್ ಅಲ್ಲಿ ಬಂದಿರೋದೆ?
ಹೌದಾ?
ಯಾರು PPP ಕಿಟ್ ಅಲ್ಲಿ ಇದ್ದಾರೆ?
I think
you can talk personally. Video.
Actually purpose is that we need to have background ಅಂತ ಹೇಳ್ತಾ ಇದಾರಲ್ವಾ.
ನೀವು ಅದು
ರುದ್ರೇಶ್
ನಾವು ಇಲ್ಲ, ಇದು ಓಕೆ.
Actually we have this way. Is this?
This is better. This is
fine.
ಈಗ ಕೂತ್ಕೊಳ್ಳಿ. ಇದು ಕೀಪ್ಯಾಡ್ ಬುಕ್ ಶೆಲ್ಫ್ ತರ ಕಾಣಿಸ್ತಾ ಇದೆ ಹಿಂದ್ಗಡೆ. ಇದು ಪರ್ಫೆಕ್ಟ್ ಆಗಿದೆ ಈಗ ಪೊಸಿಷನ್. ದೇವರಾಜ್ ಇದು ಪರ್ಫೆಕ್ಟ್ ಆಗಿದೆ. ನನ್ನ ಸ್ಕ್ರೀನ್ ಸ್ಪ್ರೇಡ್ ಮಾಡಿದಿರಿ. ಇದು ಬೇಡ. ಇದು ಬೇಡ. ಇದು ಬೇಡ, ಇದು ಬೇಡ. ಅವಾಗ್ಲೇ ಕರೆಕ್ಟ್ ಆಗಿದೆ.
ಲೈಟ್ ಇಲ್ಲ, ಅದೇ ಪ್ರಾಬ್ಲಮ್.
ಅವಾಗ್ಲೇ ಕರೆಕ್ಟ್ ಆಗಿದೆ.
ಈ ಡೈರೆಕ್ಷನ್ ಇರ್ಲಿ ಬಿಡಿ, ಈ ತರ ಕೂತ್ಕೊಂತೀವಿ.
ಇದು ಪರ್ಫೆಕ್ಟ್ ಆಗಿದೆ.
ಓಕೆ ಅಲ್ಲ?
ಕರೆಕ್ಟ್ ಆಗಿದೆ. ಇದು ಏನಂದ್ರೆ ನಿಮ್ಮ ಹತ್ರ ಒಂದು ಈ ಕಡೆ ಒಂದು ಟ್ಯೂಬ್ ಲೈಟ್ ಇದ್ದಿದ್ರೆ ಚೆನ್ನಾಗಿರುತ್ತೆ. ಈ ಕಡೆ ಫ್ರಂಟ್ ಅಲ್ಲಿ ನಿಮ್ಮ ಮುಂದಕ್ಕೆ ಈ ಕಡೆ ಒಂದು ಟ್ಯೂಬ್ ಲೈಟ್ ಇರ್ಬೇಕಾಗಿತ್ತು.
ಆ ಲೈಟ್ ಆಫ್ ಮಾಡಿ ಪ್ರೊಫೆಸರ್. ನೀವು ಇಲ್ಲೇ ಕೂತ್ಕೊಂಡ್ರೆ ಅದಿಲ್ಲ, ತೊಂದರೆ ಅಲ್ಲ. ಆ ಲೈಟ್ ಆಫ್.
ಸೂಪರ್ ಆಗಿದೆ ಈಗ.
ಏನು 2 ನಿಮಿಷ ಬರ್ತಿದ್ರೆ ನಾವು ವಿ ಆರ್ ನಾಟ್ ದ ಸ್ಪೀಕರ್. ಜಸ್ಟ್ ವಿ ಸೇ yes or no and go.
ಇದು ಕರೆಕ್ಟ್ ಆಗಿದೆ ನೋಡಿ, ಸೂಪರ್ ಆಗಿದೆ.
ಐ ವಿಲ್ ಗೆಟ್ ರೆಡಿ ವಿತ್ ಒನ್ ಸೆಟಪ್, ಡೋಂಟ್ ವರಿ. ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಬ್ಯಾನರ್ ರೆಡಿ ಮಾಡಿಸ್ಕೊಡ್ತೀನಿ.
ಏನ್ ಹೇಳಿ?
ಬ್ಯಾಕ್ಗ್ರೌಂಡ್ ಬ್ಯಾನರ್.
ಕೇಳಿಸ್ತಿಲ್ಲ.
wherever you sit and just type and sliding ಮಾಡ್ತೀನಿ ಅಲ್ಲ, ಒಂದು ಬ್ಯಾನರ್ ಹಾಕಿ
your goal
ಸ್ಲೈಡಿಂಗ್ ಚಿತ್ರ ಇರ್ತದೆ ತೊಂಬತ್ತು ನೀಟಾಗಿ ಅಂತ ಮಾಡು. ಎಲ್ಲಿ ಬೇಕಾದ್ರೂ ಅದು ಮೂವ್ ಮಾಡಿದ್ರೆ ನೀಟಾಗಿ ಬ್ಯಾಕ್ಗ್ರೌಂಡ್ ಯಾವ್ದು ಬೇಕೋ ಅವಾಗ ಯಾವ ಕಾನ್ಫಿಡೆನ್ಸ್ ಅದನ್ನೊಂದು ಪ್ರೆಸ್ ಮಾಡಿಬಿಟ್ಟು ಅಷ್ಟೇ ಅದನ್ನು ಕೂತ್ಕೊಂಡು ಮಾಡಬೇಕು. ಎನಿ ಆಫ್ ವಿ ಆರ್ ಇಂಪ್ರೂವಿಂಗ್ ಇಟ್ ನಾವು.
ಈಗ ಕರೆಕ್ಟ್ ಆಗಿದೆ. ಆಮೇಲೆ ಸ್ವಿಚ್ ಬೋರ್ಡ್ ಇದೆಯಲ್ಲ, ನಿಮ್ ಹಿಂದೆ ಒಂದು ಸ್ವಿಚ್ ಬೋರ್ಡ್ ಇದೆ ನೋಡಿ.
ಓಕೆ.
ಹಿಂದೆ ಒಂದು ಸ್ವಿಚ್ ಬೋರ್ಡ್ ಇದೆ ನೋಡ್ರಿ.
ಕೊಡ್ರಿ ಪ್ರೊಫೆಸರ್. print.
So we will set up on this thing.
ಪಾಸ್ ಮಾಡಿ. ಈಗ ನಾನು ಒಂದು PPT ಮಾಡಿ
ಒಂದು PPT ಮಾಡೋದು
ಮನೋಹರ್ 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 ಶಿಂದೆ ಅವರೇ, ಮನೋಹರ್ ಶಿಂದೆ ಸರ್ ಹೇಗಿದ್ದೀರಾ? ದೇವರಾಜ್
Hello. Hello.
Hello.
ಹಲೋ, ವಿಶ್ವನಾಥ್ ಸ್ಟಾರ್ಟ್ ಮಾಡೋಣ.
ಮಾಡಿ ಸರ್. ಎಸ್.
ಹೌ ಅಬೌಟ್ ಪ್ರವೀಣ್?
ಪ್ರವೀಣ್, ಐ ಥಿಂಕ್ he is ಅವನ ರೂಮಲ್ಲಿ ಇದ್ದಾರೆ.
Okay, I will just
Yeah, you can start this.
Okay. Shall we wait another 1 or 2 minutes?
Yeah, maybe around at 10, 20, 22.
Yes.
Hi Deva, morning.
Good morning.
ಸರಿಯಾಗಿ ಕೇಳಿಸ್ತಿದ್ಯಾ?
ಕೇಳಿಸ್ತಾ ಇದೆ.
ಓಕೆ,
wait a minute. ನಾನು ಆ್ಯಕ್ಚುಲಿ ಇದು ಎಲ್ಲ ಟು ಶೇರ್ ಓವರ್. ನಾನು ಶೇರ್ ಮಾಡಿರೋದೆಲ್ಲ ಕಾಣಿಸ್ತಾ ಇದೆಯಾ?
ಕಾಣಿಸ್ತಿದ್ಯಪ್ಪ.
Okay. I will close my sharing things.
Okay.
And then I will allow you to.
Sure.
Okay. Chat box is fine, and then people are there. Now, whoever comes, I will admit it.
Okay.
Yes. Sure. You switch on the video. Okay, ma. Fine. Yeah, you can come on video early, on early.
Sorry?
Video on early.
ಹೇಳ್ತೇನೆ.
Okay. Okay.
Fine,
that's fine. 54 members are there. Yeah, tell me. YouTube mute ಮಾಡೋದಾ?
ರಿಪೀಟ್ ಆಗ್ತದೆ.
ಸ್ಟಾಪಾ? ಇಲ್ಲ ತೊಂದರೆ ಇಲ್ಲ, ಅದ್ರ ಪಾಡಿಗೆ ಅದು ಬರ್ತದೆ. ಅದು ಸಪ್ರೇಟ್ ಯೂಟ್ಯೂಬಲ್ಲಿ ನೋಡೋರ್ಗೆ ಏನು ತೊಂದರೆ ಆಗಲ್ಲ. ಎವ್ರಿಥಿಂಗ್ ಇದಾಗುತ್ತೆ ಅನ್ಸುತ್ತಾ ಸರ್ ಆಯ್ತಾ ಓಕೆ good morning to one and all again. So we'll resume back now for the next session of this international webinar on recent trends and challenges in neuroimmunology. So here is with us our friend Dr. Manjunath MV. So he's an assistant professor in the Department of Neurovirology, NIMANS, Bangalore. So he is here with us to speak about immune response to viral infections. So
can I make a correction there? Sorry.
Yeah, tell me.
Yeah, since the theme changed from recent trends in biochemical research to neuroimmunology.
Okay.
So
yeah, that's fine.
I won't stick to what is in the title.
Okay. Yeah, that's fine. Not a problem.
I'll talk more about neuroimmunology.
Okay. I request our department faculty, another faculty, Dr. S.K. Praveen Kumar, is an assistant professor in the department. So he is going to introduce our today's right now the session speaker. Okay. Praveen?
Yeah, thank you, sir.
Yeah.
Thank you. It's an immense pleasure to introduce the speaker of the session, Dr. Manjunath MV. So Dr. Manjunath MV, who has obtained his PhD from neuromicrobiology in NIMHANS during the year 2005, followed by which he had moved to USA for pursuing his postdoctoral research in the Department of Microbiology and Immunology in esteemed institute of like Albert Einstein College of Medicine, New York. And there he was working for about like almost 5 years from 2005 to 2010, followed by which he was being elevated to associateship in research. So again, he was there for a couple of years from 2010 to 2012, the same college, that is in Albert Einstein College of Medicine, New York, followed by which he had moved on to India, wherein which he has joined as a senior consultant for the molecular and flow cytometry for the diagnostic in Anand Diagnostic Laboratories, Bangalore, for about a short span of time for about 4 months, followed by which he has obtained a position as a senior senior scientific officer at Neurovirology, NIMHANS, and there he has worked for another 5 more years from 2012 to 2017. Right, at currently, so he is working as an assistant professor, followed by which he is having an additional what do you call the responsibility, as he is a coordinator for the flow cytometry core facility at NIMHANS and actively involved in COVID diagnostic laboratories. So he is a recipient of Young Investigator Recognition Award by Bill and Melinda Gates Foundation for significant contribution towards HIV vaccine research. Okay, and he is having a US patent on incorporations of NKT cell activating glycolipid vaccine adjuvant into bacterial cell wall during his postdoctoral work. So his research interests are like immunology, immunology of infectious diseases, mainly vaccine adjuvants, diagnostic virology, immune dysregulation in neurological and psychiatric disorders. So this is a brief introduction of the esteemed speaker, Dr. Manjunath MV. So now I request Dr. Manjunath MV, sir. So kindly, you can just begin your session. So I once again welcome you, sir, on behalf of the organizer and the Department of Biochemistry. Thank you, sir. Thank you.
Thank you very much, Dr. Praveen, for your kind introduction. And I thank the organizers for having given me an opportunity to present in this webinar series. And especially excited to work along with my friend during my PhD times, but now say a long association with Dr. Devraj. So it's really a pleasure to be presenting here. Can I share the screen? Hello?
Yes.
Yeah,
Yeah.
you can.
So again, good morning, everyone. So since I just mentioned a few minutes back, the theme was changed from recent trends in biochemical research to neuroimmunology. So, my first title that I had given was immune responses to viral infections. But then to fit into the theme of neuroimmunology, I titled my talk as Immune Response, the Double-Edged Sword in Neuropsychiatric Disorders and Viral Infections. So to begin with, I would just like to give a brief introduction of about our immune system, because a lot of my slides in today's topic is going to revolve around components of immune system. So I'm sure many of you are aware of this, but just very briefly, we'll look into what are the basic components of our immune system. So you will know that immune system, or our immunity, is broadly divided into innate immunity and adaptive immunity. And you know that innate immunity is something which kicks in on exposure to a foreign antigen within first a few minutes or hours, and within the first couple of days, it's very active. In fact, it is a very efficient component of our immune system, and that's the reason why a lot of us, at least most of us, have do not anytime do not realize how many microbes we get exposed to every day. And in some cases, yes, when the innate immune system cannot really control the challenging pathogen, so then you need the help of the adaptive immunity. So what are the basic components of these two immune systems? If you look at the innate immune system, so you know all these granulocytes like the basophil, eosinophil, and neutrophil, they form an important component for the innate immune response during the first few hours and days. And they are also helped by the mast cells, and then a very important cell which has been understood very well in the last 2 decades after its discovery is a dendritic cell. So now we have quite a few subsets of dendritic cells that have been identified. And I say this is a crucial antigen presenting cell because it is very well equipped with the systems, the machinery to activate a naive T cell or a naive B cell in a very efficient manner. And it forms an integral and central component of immune responses to pathogens or in other conditions. And you have the macrophage, which again performs the antigen presenting cell function, and it is more activator and of immune system, as well as a cell which functions to remove debris during tissue repair in various organ systems. And you have the natural killer cells, which form one part of the lymphocytes, the innate lymphocytes. Okay, so the innate lymphocytes are those which cannot discriminate between varieties of pathogens, unlike the conventional lymphocytes, which we'll come to later. So these are all part of the innate immune system. And you have this interface now. So let me look at the adaptive immunity first, and then I'll come to the interface. So in the adaptive immune system, you know that the basic components are the T lymphocyte and the B lymphocyte. So the T lymphocyte mainly has two conventional types of T cells, the CD4 T cells and the CD8 T cells. And the B lymphocytes, they are involved in production of antibodies. And you know the T cells and the antibodies produced by the B cells can differentiate between millions of epitopes, millions of antigenic epitopes. And that is how we have a robust immune system which protects us against challenge of many invaders. Okay, so this is an interesting part between the innate and the adaptive immune system, that is, which forms an interface here, which includes something called the gamma delta T lymphocyte, the natural killer T lymphocyte, it's different from the natural killer cell. This is a natural killer T lymphocyte because it expresses both the natural killer cell markers as well as the T cell markers. And you have some new additions to this interface. Basically these form a bridge between the innate and adaptive immune system. So you have the mucosa associated invariant T lymphocyte. You may have heard, like some autoimmune conditions like Crohn's disease, irritable bowel syndrome, or multiple sclerosis, even rheumatoid arthritis. These cells have been implicated in pathogenesis in quite a few of these autoimmune disorders because of their secretion of huge amounts of cytokines called IL-17. And recently, we also have the innate lymphoid cells like T cells, which are again of multiple subtypes. And these cells are being understood currently by many researchers. Okay. So moving on to, so there are multiple types of CD4 T cells that are involved in various functions in our immune system. So I just would like to introduce you to only like four major types, because that would be enough for the purpose of today's presentation. So you have the Th1 CD4 T cell. Now basically a naive CD4 T cell. It differentiates under the influence of certain cytokines and by the expression of this particular transcription factor, the Tbet, into a Th1 CD4 T cell. And this Th1 kind of CD4 T cell can be involved in immune response against intracellular pathogens, as well as in like autoimmunity. Basically, it is a pathogenic T cell in autoimmunity, and this it expresses all its functions by secreting all these different cytokines. And then you have the Th2 CD4 T cell, which under the influence of IL-4 and IL-2 expresses this transcription factor GATA3 and STAT5, and all these cytokines IL-4, IL-5, IL-13. And it might, these cells are important for protection against extracellular parasites, and they're also involved in immune responses during allergy and asthma. And the naive CD4 T cell can differentiate into the inducible regulatory T cells. And you might have heard about the regulatory T cells of late quite often. So because in many conditions, the immune tolerance, which is basically maintained by these regulatory T cells, is disturbed. During homeostasis, the Tregs are highly active and they are very important. And also following an immune response to any invading pathogen, to regulate the immune responses and to bring them to homeostasis without causing any tissue damage, the Treg cells are extremely important. So in certain conditions, these Treg cells might be misfiring. Okay, and these are the CD4 T cells differentiate into Tregs by expression of these transcription factor FOXP3 under the influence of these cytokines TGF beta and IL-2. And then you have the Th17 CD4 T cell, which expresses the transcription factor RO or gamma T, which is the Th17 cells are formed from the naive CD4 T cells under the influence of these cytokines, basically TGF beta, IL-6, IL-21, and IL-23, and they secrete copious amounts of IL-17 and these various other cytokines. And IL-17 has been implicated in pathogenesis of several autoimmune disorders in the last few years. And anyhow, in terms of beneficial immune responses, they are important for protection against extracellular bacteria and fungal infections. So with that introduction, I would just like to tell you, in our department, the three major areas that we have been focusing our immunology research on are viral infections, psychiatric disorders, and immunology and neurological disorders. Under viral infections, currently we are trying to study immune markers for clinical progression in HIV, and I am personally interested in NK T cell, that is natural killer T cell agonists, which are basically glycolipids, which can activate these T cells and result in a very robust immune response by activating T cells and B cells. I am interested in the adjuvant activity of these NK T cell glycolipids against certain vaccines. At this point, we are working with this activity of this adjuvant with a rabies vaccine. And IgM responses are important for diagnostic virology to, for us to quickly detect viral infections. So I'm currently we are invested in coming up with newer ways of detecting IgM in an economical way. And this has been a long-standing interest of the department. In fact, the department is built upon this particular virus, the Japanese encephalitis virus, which is Professor Ravi's major interest for many decades now. And we are investing in understanding the T cell responses to this particular infection and vaccines against this particular virus, immune responses to the vaccines. And another component to our research, immunological research, is psychiatric disorders. So in the last few decades, although for many decades there was a speculation that the immune system is important for our health of the brain and the mind. So it was not formally proven that immune dysregulation is an integral component to several psychiatric disorders. So in the last few years, there has been a lot of support evidence that the immune system is regulated in the with our psychiatric. Hello, am I audible? Hello. Hello. Am I audible? Hello, is it there?
Yes, sir, audible.
Oh, yeah. Right. So, apart from the psychiatric disorders, we are also interested in the neurological disorders. One of them is neuromyelitis optica, which is a severe autoimmune disorder of the nervous system, which was. Oh, okay.
Little
Hello.
louder.
Yeah.
But your audio.
Okay. Is it okay now? Okay now? I'm sorry about this. Hello.
Yes, sir, you are audible.
I'm sorry. Okay, so I'll just neuromyelitis optica, which is a severe autoimmune disorder of the nervous system. We'll see it in a little bit of detail later. And how do we go about responses in these various conditions? I'd like to look at them under different, 4 different headings. So you would have heard about cytokines and chemokines, which are basically increased in several inflammatory conditions. And we classify them into a few major types, like the Th1 type, the Th2, and the Th17. Basically, these are defined by the type of cytokines. The Th1 classically involves the interferon gamma, TNF alpha, and IL-2, along with other cytokines. And then you have the Th2 type, the IL-4, IL-5, and IL-13. And then you have something called the Th17 response and IL-17. And then you have the immunophenotyping of peripheral blood mononuclear cells. Basically, we take the PBMCs and stain them with fluorescent antibodies against surface markers or sometimes intracellular markers to look at the phenotype of different subsets of immune cells, like the monocytes, the dendritic cells, the B cells, the T cells in various clinical conditions. And you also then you will go, and the next step further is looking at antigen-specific T cell responses. For example, in certain viral infections, if we are interested in what are the T cell responses targeted to, which each particular antigen, we use specific antigens from these viruses to stimulate these T cells and then detect cytokines that are secreted by these T cells. So thereby, identifying antigen-specific T cell responses. And you're all very well aware of the antibody responses against different pathogens, how they would be detected. A very commonly used method is ELISA, although you have many microbead-based assays, which have made multiplexing of antibody assays amenable and economical and and quicker. Okay. So and what is the one common technique that we use to look at the immune profiles? So the commonest technique which is across the globe used for phenotyping immune cells is the flow cytometry technique. And I'm pretty sure you would be aware of the principle of flow cytometry, wherein we are staining the cells in the blood, for example, if that is the sample, with surface markers, staining the surface markers with fluorescent antibodies, specific monoclonal fluorescent antibodies against these markers. And the flow cytometry works by acquisition of fluorescence signals from these cells when we run run the sample through a machine. So if you look at a whole blood sample after lysing the RBCs or after removing the RBCs by doing the PBMC isolation technique, so in a flow cytometer, you can look at the relative size of a cell and also appreciate the relative complexity of a cell. So by that, we can identify that lymphocyte, which is relatively smaller as compared to the other cells and less granular, internal complexity is very less in lymphocytes. So you will see these cells falling here, followed by monocytes, which are slightly bigger and more granular. And of course, the granulocytes like neutrophils are very granular and bigger in size. And you will see the distribution on top here. So this is a typical strategy of identifying your cell subsets by from the data acquired by flow cytometry. So just take you through this very quickly. When we acquire the data, this is where you plot the forward scatter or the relative size, and here you have the granularity, that is the side scatter. So based on that, we identify lymphocytes, and then you have the live dead stain, which identifies the live cells of. For most of these analyses, it's important that we analyze only the live cells. So so which means when you stain with this live dead dye, only the dead cells get stained, and the live cells are negative for that stain. And the negative cells are chosen for further analysis. Here, the example taken here is a mate cell, that is mucosa-associated invariant T cell, which is a T cell. So we have CD3, which is a pan T cell marker. And then based on the CD4 and CD8, you have the two different subsets showing up here. And mate cells are classically CD8 T cells, which express these two markers, CD161 and V alpha 7.2. That is basically the T cell receptor chain, V alpha 7.2. Based on the expression of these particular markers, we identify the mate cells. So this is just one example about how we we identify different cells. And multiparametric flow cytometry is a very common technique these days used across the globe for identifying many different subsets of cells in our peripheral blood. And you also can look at T cell activation, T cell exhaustion, okay, and you also have a particular panel that has been standardized for identifying regulatory T cells and the mate cells. I told you just now, the one panel to identify natural killer cells, and then you have a panel to identify the T cell function by looking at intracellular cytokines. So this is basically, these are like the lasers that excite all these different the blue color here represents a blue laser and a red laser, and this here represents a violet laser. These are the dyes excited by these lasers when we run the sample through the flow cytometer, and we have detectors for this particular fluorochromes. These are all different fluorochromes which have been used to label the antibodies. And when it comes to serum or plasma biomarkers of immune activation, there can be of different types. Activation markers like soluble CD14, soluble CD38, which you will be aware of. A few cytokine receptors, a plethora of cytokines and chemokines that are detected in different conditions. And you have others also which help you identify inflammation. Okay. So just to look at what we have done with peripheral immune alterations in psychiatric and neurological disorders. Of course, it is very difficult to look at the CNS itself. The central immune alteration is very difficult to look at. Maybe we have some CSF in certain conditions, but not all conditions. But it's very difficult to work with a small amount of CSF to understand immunological alterations, although that would be the ideal sample. Okay. So you would be aware of the fact that the central nervous system was considered a immunologically privileged site. That means wherein the representation of the immune system is very minimal or almost absent. That was the thinking for a long time, for many decades, about the central nervous system. But one remarkable paper in Nature in 2015 kind of changed this opinion and showed evidence for, and the previous thinking was mainly because of the understanding that there was no lymphatic drainage directly from the CNS. So there was no way how the immune system could quickly see a presence of a foreign antigen or a particle within the CNS. So the understanding has changed over the last few years, and this paper showed there are actually structural features of lymphatic vessels in the central nervous system. So here it shows a mouse brain with labeled, the green color is the labeling of the lymphatic vessels on the surface of the mouse brain in the meninges. So from a previous, because of those discoveries, and also this was identified in human autopsy brains, these structures. So based on that understanding, so this used to be the old picture, and this here is updated picture which reflects the new discovery, showing there is lymphatic drainage from the central nervous system. So this now actually has opened up many possibilities for immune mediated interventions for several CNS disorders. Okay. So let me just come up with the first example of the disorder that we have looked at. It's called postpartum psychosis. So this was done with our clinical colleagues from psychiatry. So postpartum psychosis is a severe psychiatric disorder affecting new mothers, about 1 to 2 per 1,000 births. Depending on the geographical area, this might vary. The number may be a little higher. This basically is probably now Western data. Okay, it's defined as an acute episode of mania or psychosis developing shortly after childbirth, typically within the first few weeks. So women classically present with frank psychosis, including hallucinations and delusions, mood lability, perplexity, and confusion. I'm pretty sure a lot of us would be aware of new mothers, not really feeling normal, in our households, and it really takes a long time for them to get back to normal. And it's pretty imaginable, like because of all the changes that happens during pregnancy, huge variations in hormones, which can affect all the different organ systems, including the immune system. If the hormones and all the other various other physiological changes do not return back to the normal quickly right after delivery within the first few days, then there might be some aberrations which could lead to postpartum psychosis. This is one picture where, like yesterday, you would have heard from Professor Ravi how the HPA axis, the hypothalamus, pituitary, and adrenal axis, and stress would, these are involved in hyperactivation of the immune system, which can have many devastating impact on the body. So this is one such example during postpartum depression or even postpartum psychosis. So this axis is involved in upregulation or of several neuroinflammatory molecules in the body. Now these have been shown during the postpartum period after delivery in mothers with postpartum psychosis, that some of these neuroinflammatory markers are expressed in high levels, like some cytokines like IL-6, the TNF, the IL-17. There have been multiple studies to show that the immune system is in fact altered significantly. including ours. So in the prepartum period, the cytokine levels or the inflammatory markers are in a regulated at regulated levels. But in this particular condition, postpartum psychosis, the cytokine levels and other inflammatory markers are highly upregulated. So there is disruption in the crosstalk between stress hormones and neuroinflammation, which may contribute to postpartum depression or postpartum blues is what is called, or psychosis. So no, as I told you, we can work from the peripheral system, and also currently the thinking you would have heard that a lot of these triggers that lead to alterations in the central nervous system starts in the periphery. In fact, from the gut, right? So all of us are now familiar with the gut microbiome and so much of research going into that. So the thinking is that several of these psychiatric disorders and neurological disorders, the trigger happens from the gut, where there is a dysregulated immune response starting from there. Some cells are triggered in an untimely fashion, and they result in high levels of inflammation and finally resulting in a psychiatric disorder here. So we were working with peripheral blood. So that is what we are trying to look at, the PBMCs. So which has all these different cells. CD4, CD8, B cells, the Tregs and the cells representing the, hello? Hello?
Wait a minute, Manju. I will unmute all. Sorry, mute. Okay, continue.
So yeah.
Unmute.
So we are working with the peripheral immune system. Basically, all these constitute the peripheral immune cells here in the blood from the adaptive immune system, and you have the innate immune system represented by NK cells, dendritic cells, monocytes. So we would like to see whether these are upregulated or downregulated, and how exactly they may be impacting the brain, resulting in these disorders. So in this effort, in this study that we did a few years back, so we found that when we, based on flow cytometry phenotyping, we found that a few types of monocytes, especially the non-classical monocytes, which are known to be regulatory in function, which are known to bring about resolution of inflammation. Okay, after immune response to a pathogen or during injury. So these particular monocytes are decreased in postpartum psychosis. Here, the black bars show the postpartum psychosis, percentage of cells in the condition as compared to the normal uh, uh, healthy individuals in the gray bar. Okay, so that is one important finding that we found. So probably the immune system is not well regulated. It's an indication. And you also have these dendritic cells, the myeloid dendritic cells or the MDCs, again, which are decreased in postpartum psychosis here. So actually, if you look at it, the myeloid dendritic cells are supposed to be pro-inflammatory, but we have seen a decrease, a significant decrease in those cells. So it's very difficult to understand how exactly the different components of the immune system, under the influence of this, huge physiological changes occurring in the new mother, how the immune system is being dysregulated. This shows that immune system is dysregulated. There's some evidence here. Okay, that's about the monocytes and the dendritic cells. And with respect to natural killer cells, again, so the first set of graphs here, the first pair, again you see these are the classical natural killer cells which have the cytotoxic function, which kill virus infected cells, or which can eliminate cancer cells in our body. So these are decreased. I mean, basically they have a protective function, and these are decreased during this postpartum period. And that is understandable because the immune responses are quite suppressed during pregnancy is what is classical understanding. So these may have been taking time to come back to normal. And so that is one thinking. But you also have these regulatory NK cells. These are called the CD56 bright natural killer cells, which are decreased again in, which are increased in postpartum psychosis. So when it comes to the natural killer cell compartment, it looks like, it is a beneficial response in postpartum psychosis. But this may not be enough to regulate immune responses. And if you look at the adaptive immune cells, we see a huge decrease in naive T cells, and there's an increase in activated T cells, and there's also alteration in memory Treg population. Actually, the memory Tregs are increased in postpartum psychosis. Again, this for us is a good picture to see. Like memory Tregs should be increased in pregnancy so that to regulate the immune responses which may go haywire. But yeah, we cannot fit in all this into the puzzle and make the understanding easy. But that is what we have got. So basically, we have seen alterations in both innate and adaptive immune arms in postpartum psychosis. Okay, so one hypothesis, or I wouldn't say hypothesis now because there's a lot of evidence already for this hypothesis. It's several psychiatric disorders, as I told you, is now speculated to be resulting from a dysregulated immune system. And basically, maternal immune activation, when the embryo is developing, when the fetus is growing in the mother, gestating mother. For example, if the mother suffered from some infection and that resulted in immunological activation, so that the immune response, which results in cytokines like IL-6, and that causes activation of Th17 cells, which release a lot of IL-17. This is shown to impact the cortex formation of the developing fetus, and basically affecting neurodevelopment in the very early stages, which is speculated to predispose the baby for several psychiatric disorders following birth. Okay. So this is the working hypothesis now. So during immunological activation of a mother, who's a gestating mother, due to either autoimmune conditions or infections, or there could be a genetic predisposition, which may result in IL-6, resulting in activation of Th17 cells. They release a lot of IL-17. As we saw earlier, it results in maldevelopment of the brain of the fetus. So, and then during childhood, the fetus, the child who was exposed to maternal immune activation when inside the mother, may be predisposed for developing autism spectrum disorders or other psychiatric conditions, which we'll see later. So even if the child during childhood, if if there are no presentation of any clinical manifestations, so during adolescence, the offspring which has been exposed to maternal immune activation is more susceptible to second hits such as stress and drug abuse, and may show up psychiatric disorders or neurological disorders during adulthood. Okay. So moving on to the next psychiatric disorder, where we try to understand immune dysregulation, that is autism spectrum disorder. So it is a neurodevelopmental disorder characterized by two main signs: multiplicity, social interactions and communication, and repetitive or stereotyped behaviors. So a lot of us would be familiar with what autism means and how difficult it is, for the child, and more importantly for the parents, to cope with child, or to take care of the child with autism. So some of the evidences for immune aberrations in autism spectrum disorder include postmortem findings in ASD brains. There is evidence of chronic brain inflammation, the microglia, which are innate immune cells of the brain, which form, which serve as antigen presenting cells, as well as cells which are involved in tissue repair, which are involved in priming of the developing neurons in the fetus. So these are activated, and there is evidence for increased levels of inflammatory cytokines, and there is altered blood-brain barrier. I mean, you would know blood-brain barrier is highly selective, and we need a very healthy blood-brain barrier for for a healthy individual. And if this is altered, it can cause havoc. And so people have shown peripheral immune dysfunction, like taking out blood cells and looking at different innate and adaptive immune cells. So they've shown activated monocytes, dysfunctional NK cells, dendritic cells are increased, altered T cell profiles, and you can have cytokines, autoantibodies targeting several brain antigens, etc. And it's also seen that some of these a lot of these individuals have other comorbidities like allergies, asthma, type 1 diabetes, GI dysfunction and inflammation, celiac disease, gut dysbiosis, all these pointing out to a dysregulated immune system, which is probably not well developed, or some events during the embryonic stage affected the development of the immune system as well as the nervous system. So in our efforts, again with our psychiatric colleagues, as part of a PhD project of Dr. Salah Bashir, we looked at children with autism spectrum disorder, and we found that the cytokines which are implicated in pathogenesis of an autism spectrum disorder, at least in the animal models, these were elevated in the children with autism spectrum disorder as compared to the controls. So here you see IL-1 beta, although there is a trend for increase, we didn't find a significant elevation. But with IL-6 and IL-17A, which are pro-inflammatory cytokines, or which can be referred to as pathogenic cytokines, were elevated in these individuals. And similarly, we looked at the Th1 T cell types and Th17 T cell types, which are pro-inflammatory in nature. We found that the Th1 and Th2, again, there was a trend for increase. We did not see a significant increase, but activated Th17s were significantly elevated in the children with autism spectrum disorder. I'm sorry about this typo here. That's supposed to be Th17 cells. So there's an elevation of pro-inflammatory Th17 cells that we observed in this. And coming to another important type of T cell, I mean that innate immune cell, that is a dendritic cell. I told you the myeloid dendritic cells, they are pro-inflammatory in nature. And we found that these cells were significantly elevated in these autism individuals, the children with autism spectrum disorder. So because of these alterations found in these children and in several other psychiatric disorders, so in the last many years, there have been attempts to actually, so there is some disturbance, Deva. Hello? Hello? There is some disturbance, sorry.
Hello.
Yeah.
Yeah, just I will mute them. I think some people. Yeah, just I'll ask you to unmute. Yeah, please. Is it fine?
Yeah, is it okay? Hello? Is it all right?
Yeah, it's fine.
Okay. So that was, in summary, we found alterations in a few of these immune cells in autism spectrum disorder, and actually the findings were in fact concordant with what has been described in the field, and well supported in a mouse model of maternal immune activation, followed by the embryos showing behavior like ASD in humans, like the babies, the pups showing ASD like changes in their brain and in behavior. So that's about autism spectrum disorder. And another psychiatric disorder that we looked at is schizophrenia. and you're all very familiar with this disorder as well. So it's a serious mental disorder in which people interpret reality abnormally. So it may result in some combination of hallucinations, delusions, and extremely disordered thinking and behavior that impairs daily functioning and can be disabling as well, and highly stressful for families who have to take care of these individuals. So people with schizophrenia require lifelong treatment. Early treatment may help get symptoms under control before serious complications develop and may help improve the long-term outlook. So this is another disorder where, so I was just trying to tell you before before the break, that currently in the field, in fact, clinicians, psychiatrists are looking at immune-based interventions like anti-inflammatory drugs. Can they improve symptoms? I mean, can they improve these patients who have psychiatric disorders? If immune system is really involved in these in the pathogenesis, then it should actually help in alleviating suffering due to these psychiatric disorders. And to tell you, it's not so straightforward. So the question comes back, is it actually the immune system which primarily leads to these disorders, or is it the other way around, some other neuroendocrine systems being imbalanced or dysregulated that leads to these immune system immune components being dysregulated? So it's not so straightforward. There have been some encouragement from some studies, but still not there to use immunological interventions for psychiatric disorders. So what are the different abnormalities that you could see in schizophrenia? So it might be that the microglia which are activated in the CNS, or some monocytes within the in the periphery, that may lead to pro-inflammatory cytokines or anti-inflammatory or decrease in anti-inflammatory cytokines, which can affect some of the main pathways involved in immune system activation. One of them is kynurenic pathway. Kynurenic acid is a breakdown product of tryptophan. So that is known to result in immune activation, innate immune activation. This activated microglia are ultimately going to lead to higher breakdown of tryptophan, really causing an increase of kynurenic acid, and that results in immune activation. And these activated microglia, as I told you earlier, are important for neurodevelopment. So they are important for synaptic pruning during neuronal development. So that might be affected. So, and it can also activate T cells, and so which ultimately the CD4 T cells can result in activation of B cells, and resulting in autoantibodies. So that might again result in an insult against the brain, finally resulting in neurodegeneration and altered neurodevelopment. And there is something called the positive symptoms, negative symptoms, and then you have the cognitive symptoms, where all these different functions are affected due to microglial activation. So in our efforts, we saw looked at peripheral immune activation, and just to not to make this very complex for you, we looked at the Th17 cells. What is the level of activation of these Th17 cells in patients with schizophrenia, okay, as compared to healthy controls? And you have the mate cells. I told you mate cells have been shown to be important in autoimmune phenomena. Schizophrenia is being thought of as an autoimmune disorder by some of the researchers. So we looked at these, and we found that these T cells, the Th17 cells and the mate cells, are in fact increased in patients with schizophrenia. So, and then we also found that people in the field have proposed this idea wherein if the Th2/Th17 ratio is balanced, then the immune system, that means the immune system is under homeostatic conditions without any dysregulation. But in schizophrenia individuals, we found that there is an imbalance in the TH17, TH2 to TH17 ratio, basically suggesting that the TH17s are increased as compared to TH2, which are believed to be protective in schizophrenia, again resulting in a pro-inflammatory condition. So that's about schizophrenia. I'll move on to a neurological disorder, neuromyelitis optica, which I told you was initially for many decades back was considered a variant of multiple sclerosis. But then the identification or discovery of a immunoglobulin, IgG, which reacts against our water channel protein aquaporin 4. That showed that this disorder is very different from multiple sclerosis, and even in the clinical presentation and radiologically, people, the clinicians see there are a lot of unique features for neuromyelitis optica as compared to multiple sclerosis. So basically, it's an inflammatory disorder of the central nervous system characterized by severe immune-mediated demyelination and axonal damage, predominantly targeting optic nerves and spinal cord. So this disease-specific serum antibody, the NMO-IgG, is used for diagnosis as well. If an individual who is showing clinical symptoms which are related to neuromyelitis optica, and if this antibody is detected in the blood, it's diagnostic of neuromyelitis optica. So this is the proposed mechanism of how individual could develop neuromyelitis optica in a stepwise fashion. So as I told you, where exactly the trigger happens, how the immune system is dysregulated, finally resulting in a neurological disorder. The trigger factors are unknown as of now, although there is a lot of suggestion that it might start from the gut. The dysregulated gut microbiota could be one of the important reasons. So with the unknown trigger factor, there is breaking of tolerance. So tolerance means the immune tolerance. I told you the Treg cells are predominantly involved in maintaining immune tolerance. So if that is affected, then there might be activation of unnecessary activation of the immune system, and then there could be granulocyte recruitments, and then you have activation of T cells, B cells. The B cells produce high levels of aquaporin 4 IgG, the immunoglobulin against the water channel protein, and these antibodies are shown to cross over to the CNS from the periphery and bind to the water channel protein, and that leads to the activation of the complement system, thereby bringing about lysis of several cells of the central nervous system. And you can also have the neutrophils and eosinophils, which can enter the brain, bringing about causing a lot of damage. And the macrophages, which enter the parenchyma, can result in myelin phagocytosis and demyelination, finally resulting in the clinical picture of neuromyelitis optica. So basically, it involves the lot of immune cells from the periphery and the antibodies crossing over the blood-brain barrier towards the central nervous system and causing this damage. So here you see the complement-mediated activation and lysis of the astrocytes. So as a part of Dr. Hari Krishna's PhD project, we looked at what are the cytokines and some soluble receptors of these cytokines which might be altered. We actually, in fact, looked at several of these, and we found that some of these, like the IL-10, the TNF. So here RRMS refers to the multiple sclerosis patients, relapsed remitted multiple sclerosis. So we tried to compare the immunological picture of MS with, and as I told you, the NMOSD and multiple sclerosis are very different disorders with a specific signature. We wanted to compare these two different, but both are autoimmune disorders. We wanted to compare the picture of NMO with MS and look at the specific differences. In fact, we found that some signatures like the IL-10 here were very specific to NMOSD. It was highly elevated, and you have the TNF receptor 2, which was highly elevated in NMOSD. And in contrast, the TNF receptor 1 was not very altered in NMOSD, but it was very suppressed in multiple sclerosis. And when you look at TNF alpha, it was upregulated. Both I told you are pro-inflammatory conditions, both MS and NMOSD. And you see that TNF, which is a signature of inflammatory response, is elevated both in MS and NMOSD as compared to the healthy controls. And similarly, you have other pro-inflammatory cytokines like the IL-17A, interferon gamma, IL-6, and you also have IL-4, which is a Th2 cytokine, which is highly elevated in neuromyelitis optica. So basically telling us that it's a condition where there is high level of inflammation, which can be seen even in the peripheral blood. Okay. So you also have CD8 T cells, which are highly activated. You can see that very specific to NMOSD, these CD8 T cells are elevated. So, but the antigenic targets of these CD8 T cells are not known. Only the antibody of NMO is known to bind to aquaporin 4. And you also have elevated Th17 cells. So you might have noticed that I've been coming back to Th17, the psychiatric disorders and neurological disorders here. So the Th17s seem to be very, all these different pathways seem to converge to elevation of Th17. So yeah, it is difficult to believe, but that's the data we have. So what is the trigger and what is the final phenotype? What results in this final phenotype is something which we need to investigate further. So regulatory Tregs are, I told you Tregs are important, and these Tregs are decreased in neuromyelitis optica. So again, a sign to show that the immune system is not well regulated during this particular disorder. So am I okay on time? Should I continue? Hello, David.
Yeah, you can continue.
Okay. So that brings us to the end of the neuroimmunology aspect of it, as with respect to what we have done with psychiatric and neurological disorders. So I'll just try to shift gears here and move into the areas of the research area of our own department, primary research area, that is viral infections. So you all would be familiar with this. When during a viral infection, there will be two components to the immune response, that is the innate immunity and the adaptive immunity. And yesterday, Dr. Ravi, while talking about COVID-19, pointed out to you the virus is very good at suppressing the innate immune response. He was talking about these particular cytokines, the type 1 interferons, which are crucial for protection against viral infections during early stages. Okay, the interferon alpha and beta. So during a typical innate immune response, these interferons go up during the first 2 to 3 days and immediately come down. And then one of the important functions of these cytokines is to recruit the next arm of the innate immune response, that is the NK cells or the natural killer cells, which act against virus infected cells. So these come in and peak at about day 3 to 4, and then they go down. And then and one important function of these innate immune responses is to recruit the adaptive immune system in a very efficient manner. Okay, so the innate immune system, which for long was thought to be not very important, because of our success with vaccines, which hello, Deva, there is some disturbance. Sorry.
1 minute. I will hold on. I will. Yes. You can unmute and start.
Yes.
Yeah, thanks.
Still there, Deva.
People are talking?
Yeah.
I think okay. Yeah, you can continue.
All right. So yeah, so it's very important for the innate immune system to hand over the signals, hand over the baton in a very efficient manner to the adaptive immune system. So the adaptive immune system kickstarts efficiently to control the invading pathogen. So in that part of the immune response of adaptive immunity, we have the virus specific cytotoxic T lymphocytes.
Viral infection. But not adaptive. Adaptive comes down.
Hello, sorry.
Sorry. Oh, sorry.
Can I continue, Deva? Sorry.
Yeah, please.
All right. So basically the CD8 T lymphocytes or the cytotoxic T lymphocytes are crucial for protection against viral infections. So these form an important component of the adaptive immune response. Of course, we need the CD4 T cells. They are the helper T cells which drive the CD8 T cells and other cells of the immune system to control the invading pathogen. So, and then another crucial component is the antibody, especially the neutralizing antibodies, which take care of the virus. So as a result of this successful innate and adaptive immune response, you have the virus titer going down within the first 2 weeks after the infection. And in fact, because this particular component of our immune system is so efficient, we don't even realize how many pathogens we encounter every other day. So the immune response against pathogens can be broadly divided into 3 types. Type 1 immunity, wherein you have one particular transcription factor, which I introduced very early on when talking about the types of CD4 T cells, the T-bed transcription factor resulting in a cytokine, interferon gamma. So there are other cytokines as well involved, but this is a classical signature of type 1 immunity. This works against intracellular pathogens, including viruses. And pathogen clearance is through effector cells, including the innate lymphoid cells, which are recently discovered, the natural killer cells, the cytotoxic T lymphocytes, and you also have the Th1 cells, the CD4 T cells, basically the Th1 cells, which a subset of them is known to directly function as cytotoxic T lymphocytes themselves. Although traditionally CD4 T cells are known to be helper T cells without a direct effector function. Okay, so some of these cells, including COVID-19, now people have showed they have a cytolytic function, the CD4 T cells. They directly go and kill infected cells. The second type of immunity is a type 2 immunity, which involves the transcription factor expression of, that is GATA3, resulting in cytokines IL-4, 5, and 13, and also expression of this particular immunoglobulin. And they are particularly important for defenses against worms like helminths. And primarily the pathogen clearance is through epithelial cells, mast cells, eosinophils, and basophils. Okay. And then you have the type 3 immunity, which involves expression of the transcription factor Rho or gamma T, basically resulting in differentiation of CD4 T cells into Th17 cells. And these cells secrete a lot of IL-17, IL-22. And then you also have the ILC3, the innate lymphoid cells 3, which perform a similar function of secreting IL-17. And these are important for protection against fungi and extracellular bacteria. And the pathogen clearance is mediated by neutrophils here. And one important condition, like where these type 3 immunity is implicated in pathogenesis, is autoimmunity, like I told you earlier. Okay, so I just I have to offer the flavor of the season, although I titled my talk as neuroimmunology. So unless I touch on COVID-19, my talk would not be very relevant. So I thought I will we'll just look at briefly what are the immune responses that occur in COVID-19 and what is the understanding that is currently available. So in a typical immune response, if everything is going fine, so when there is an infection, there will be SARS-CoV-2 replication in the upper respiratory tract, and there will be endocytosis of the virus by a lot of cells, including the antigen presenting cells, that is the dendritic cells. I told you the dendritic cells are key cells for eliciting a good immune response. So when there is antigen presentation by a dendritic cell to a CD4 T cell or a CD8 T cell, okay, so there is activation of CD8 T cells resulting in high levels of proliferation, expansion of these CD8 T cells. There is also secretion of inflammatory cytokines, resulting in viral clearance. The viral clearance can be mediated by different pathways. It could be either apoptosis of infected cell through the perforin granzyme or through the TNF mediated, fast ligand mediated apoptosis of infected cell, or they could also result in pro-inflammatory mediators, which actually result in recruitment of other important immune cells and result in elimination of the virus. So thereby there is clearance of the SARS-CoV-2 from the CD8 T cell compartment. Now, if there is activation of CD4 T cell, again, the CD4 T cells activated, they proliferate, and finally their function is to activate B cells. The B cells result in secretion of antibodies, which result in clearance of the virus. So this is a very successful immune response, which probably is occurring in majority of the individuals. As we know, COVID-19, asymptomatic individuals or individuals with milder disease actually predominate as compared to those with severe disease. So some understanding that is available is, so during initial stages of the infection, when people looked at peripheral T cell counts, they're highly increased, they're activated, the CD8 T cells, or they're expressing their effector molecules like granzyme B, perforin A, which are important for cytolytic killing of infected cells. And CD4 T cells are highly activated and proliferating. They secrete a lot of cytokines. And then you have the regulatory T cells, which are also secreting IL-10. But as we progress towards later stages of the infection, there is high levels of inflammation, and also it results in severe disease. And these cells, or these different cells, are depleted in the peripheral blood. And these are the phenotypes of these T cells, which are, they're expressing high levels of activation markers, and you also have a lot of cytokines being secreted. Basically, they are not able to rein in, or they're not able to control this immune mediated activation, and they go on secreting a lot of chemokines and cytokines, which result in recruitment of different other damaging cells to the different tissues. And another theory, this is with respect to cells, when it comes to antibodies. So this is a typical picture of how following infection, the viral RNA goes down by 3 weeks. And then you have the, with respect to immune response, very early on, you have the IgM antibody coming in. And then you have the IgG antibodies, which are more important for long-term protection. And there has been some proposals that have a speculation that in some critically ill patients, the IgG antibodies may actually be causing a enhancement of the disease. That is, these IgG antibodies, instead of being very efficient in their neutralizing function, they may be suboptimal, which actually results in viruses being taken up by the cells through these antibodies, thereby increasing the rate of infection, and also facilitating viral replication in the host. So there is no much understanding, but some pointers show to the role of these suboptimal neutralizing antibodies in the pathogenesis of COVID-19. Okay, and there's still, we don't have very good understanding about how efficient the memory B cells are. So you might have read in the news or in articles, sometimes people saying there is no immunity to reinfection, and some people saying there's actually immunity to reinfection. So the picture is not very clear. So the antibody dependent enhancement was what I told you. You might have heard that in the context of dengue. This is a very well understood phenomenon in dengue. So that is being now also, people are saying that the suboptimal neutralizing antibodies against other coronaviruses, which might be facilitating ADE of COVID-19, resulting in lung pathology. Okay. So this term again is it's kind of cliche now. Many people are very familiar with this term, cytokine storm. Basically, it means high levels of cytokines being secreted in the lung. That is because the virus suppresses the immune system early on. So the immune system, our body, in an effort to control this virus, tries to bring in more and more soldiers to one particular tissue, and it's not able to be efficient, but goes on secreting a lot of cytokines and chemokines, and that results in tissue damage. So this is what I was referring to. For example, in the lungs, so a lot of cytokines and chemokines, which bring in all these different cells, lymphocytes, dendritic cells, macrophages, and resulting in tissue damage. So immune dysregulation is the hallmark of COVID-19. So, and you have seen yesterday from Professor Ravi's talk, how type 1 interferons or the innate immune response is suppressed. So this is all the evidence to show that, like what he said yesterday, there is more to COVID-19 than the virus. So all this is the evidence. So in fact, I'll just share with you one very elegant paper which recently came out in Nature, which was published by a group from Yale University, US. So they took a set of 113 COVID-19 patients with moderate and severe disease. So they found that in moderate disease, early increase in cytokines was seen, but there was a progressive reduction in type 1, that is antiviral cytokines, and type 3 responses, which are, and when it comes to severe disease, they found these elevated responses of cytokines that sustains through the course of the disease, and it is accompanied by increase in type 2 kind of response, which includes IL-5, IL-13, IgE, and eosinophils. So based on this, they said there are 4 immune signatures which decide the disease trajectories, how exactly the disease progresses. So that the 4 immune signatures have been divided into that each, the A being growth factors, B being type 2 and type 3 cytokines, and C being a mixed type where you have all 1, 2, 3 type of cytokines, and the D, chemokines involved in leukocyte trafficking, including all these different chemokines. Okay, so this is one picture where you can visibly appreciate. So these are all you can see on this axis here. They have looked at some 70 odd different cytokines and chemokines, like basically immune markers in people with early and late stages, both in people with moderate disease and severe disease. So you can see that the early increase in cytokines in people with moderate disease, it comes down here, okay? And the early picture is similar to both people with moderate and severe disease, right? The early picture is similar to both these groups. But when it comes to late disease, so the people with moderate disease, the cytokine levels are in fact decreasing as they improve. But in people with severe disease, you see these high levels of expression. So this matrix shows you a lot of these different cytokines and chemokines, or the inflammatory markers, remain elevated during late stages of the disease. Okay, so with that, I would like to move to what is happening in the vaccine development scenario and how we are trying to exploit immune responses against these different vaccine antigens. So you might be aware there are different approaches to making this vaccine. And yesterday you saw that the S protein is the most important protein for the virus.
Manju, how long it will take?
Sorry, another 2-3 minutes. Is that okay?
Fine.
Okay. So the spike protein, so the receptor binding protein of the S protein is the most important. So there are different approaches to making these vaccines, basically expressing the S protein by different approaches, either it's RNA vaccine, DNA vaccine, recombinant, there are a lot of vaccines which are in different stages. Hello, can I continue?
Continue, Madhapa.
Can I continue? Okay. So how do these different vaccine approaches, what are they looking at? Like how do they plan to elicit immune responses? Basically, all these different vaccines, whatever approach they are taking, whether it's a whole cell inactivated vaccine, a virus vector, a different virus expressing SARS-CoV-2 S protein, or a subunit vaccine, or a virus-like particle, or a DNA-based or RNA-based vaccine, they will need to target dendritic cells. Through the dendritic cells, there is activation of B cells, the CD4 T cells, and the CD8 T cells. And you have all these different T cells now and B cells, which are educated to respond against a pathogenic challenge. So the B cells respond to the neutralizing antibodies. All these are resulting in blocking of the replication of SARS-CoV-2 virus or blocking of infection itself by the neutralizing antibodies. So, all right. So, because this is, the theme is on neuroimmunology, I just thought I'll wind up with, how SARS-CoV-2 is being thought of, what is its role in CNS manifestations. Now, whether are there CNS manifestations? People have reported, neurologists have seen quite a few cases across the world with neurological manifestations resulting from COVID-19 infection. And this paper review has proposed that there could be 4 different ways how COVID-19 or SARS-CoV-2 infection could be affecting the CNS. One is by cytokine mediated cell damage of the neurons. Basically, the SARS-CoV-2 is entering through the olfactory neurons from the olfactory bulb, from the nasal cavity, and gaining access to the brain is one of the proposed hypothesis. So it could be direct cell damage to the cell damage through cytokines, or direct damage of the cells by entry of the cell through the blood through the virus through the blood-brain barrier into the brain parenchyma and resulting in damage of the brain cells. And then you can have auto antibody like phenomenon where the virus drives the synthesis of antibodies by B cells, which are could have an autoimmune phenomenon, result in autoimmune phenomenon causing damage of the CNS. And then the stress itself due to COVID-19 infection and all the associated factors, which might result in the damage, like what you saw yesterday, how it results in the dysregulation of the HPA axis and causing psychiatric symptoms. Okay, so I'll end with this. This is just to show how the virus could be gaining entry into the CNS through the olfactory bulb, or it could directly infect the, go through the blood-brain barrier from the blood, from the bloodstream into the brain parenchyma by infecting the endothelial cells and crossing over to the brain parenchyma. So this is what happens here when it crosses over to the blood-brain through the blood-brain barrier into the parenchyma. Okay, so I think I will end with that. This is something which we have, because with what are the other viral infections that we deal with. So I wouldn't continue further. I think we have discussed enough. So I would like to acknowledge all the PhD students from our department, Dr. Gopinath, the MD Psychiatry, basically, Dr. Mahesh was a postdoctoral fellow, Dr. Salah Bashir, Varun and Hari Krishna, all PhD students. And I would like to acknowledge Professor Ravi, who is my mentor, and for all the support, and all my departmental colleagues. So with that, I will take questions. Thank you. Thanks for your kind attention and patient listening. Can I stop sharing, Deva?
Yeah, you can stop sharing.
Right.
Yeah.
Sorry, I hope it was not.
No, that's fine, not a problem. Okay, because we have another meeting, right? Faculty of Science. Okay, just I will thank you, Manju, for coming over here and delivering the talk.
Okay.
Particularly to address the role of immune system in psychiatric disorders and postpartum period, ASD, schizophrenia, neuroleptic optica, and so on. Defense of our viral infections too.
Yes.
So now you can take some more questions and all. Okay, I would just thank you before I leave.
Right, sure.
Yeah.
Thank you, Deva. Thank you for the.
I will catch you later.
Dr. Praveen.
Okay. Yeah. So please, address the questions, and probably they would be giving, in the chat box.
Right.
Dr. Praveen will be here around. So
Right. Sure.
he
Thank
will
you.
continue.
Thank you, Deva.
Yeah.
Right.
Yeah. Yeah.
Thank you.
Bye. Bye.
Bye.
Hello, sir.
Hello. Yes, please.
Sir, one question.
Yes.
Sir, what is the best treatment for the autism, ADHD?
No, there is no targeted best treatment as of yet. It's basically by, mostly by psychiatric, and psychological interventions. No, there is no drug as such.
Okay.
To treat autism. There's no
Sir,
specific.
one more question about there were reports that blood group A people are susceptible to COVID-19. Some reports were there.
Right.
Is it true, sir, or
Any idea why, what was the reason behind that? Any idea? Because there's no evidence to show that. There's no clear evidence or enough evidence to show that.
yes, sir. Some reports were there. Some China people they told this blood group are more susceptible.
Yeah, no, there have been several such reports last 4-5 months,
Right.
implicating many different reasons. So nothing has really stood the test of time.
Okay, sir. Okay. Thank you, sir. Thank you. Thank you. It was a nice presentation.
Thank you. Thank you.
Sir, I have one question.
Please.
So this is Dakshayani.
Yes, please.
So what is the percentage of transferring the psychological disorders from parents to offspring, sir?
Good question. They say there is a good possibility. Yes. If there is familial history, the susceptibility increases.
Okay.
I won't say it's 100%. Yeah, but they're prone, depending on other factors, right? Like environmental factors. If there are other triggers, then they become more susceptible. For example, in postpartum psychosis, women before becoming pregnant, if they had bipolar disorders, during pregnancy, they are shown to be more susceptible to this particular disorder. Yeah. So there is an association. Yeah. Okay. I hope I answered your question. Any more questions? I hope you found the crowd found it relevant for the topic. So. Any more questions or?
Any more questions from the audience participants actually?
So I see that somebody has asked what is the function of CD8 T cell in COVID-19. I think we kind of looked at it very elaborately. All these T cells are activated conventionally. So it's from one Mr. Anup, the question. I read that. So CD8 T cells are important for protection, but the timely response is what is essential. And the virus seems to be gaining an upper hand in at least in people with severe disease. But in people with moderate disease, the CD8 T cells should have the known functions like clearing of infected cells quickly. So they have the cytotoxic T cell function.
Okay. Yes. Anyone like? Is anyone
so there is HPA axis activation leads to cortisol, which is anti-inflammatory in nature. I mean, it's not as straightforward as that. So people under stress, who have high levels of cortisol also show high level of activation, immune activation. So you can't just term activation, cortisol as totally anti-inflammatory. High levels of cortisol can also lead to high levels of immune activation.
Okay.
What is the role for co-inhibitory activator in COVID? So I didn't understand the question. What is Mr. Anup, co-inhibitory activator? Can you please speak?
Yes. Yes, Mr. Anup, are you here?
Okay.
Please. Okay.
All right. Can I move on to the next question?
Yep, sir.
how the child will be affected if the mother is affected with COVID.
Probably, I
Okay,
think you answered
good.
the same question.
So, yeah, I mean, if there is high levels of activation in the mother during pregnancy, as I told you, there could be a possibility the child might become susceptible to, you know, maldevelopment of the neurons, you know, when before birth. It's possible that you know, they might be susceptible to other disorders after birth during childhood or adulthood. I mean, but there is no evidence yet because it's only like 4 or 5 months since all this started. So only time will tell, you know, how much of a problem is COVID-19 infection during pregnancy to the child. So, right. What are the inhibitory cell responsible to COVID-19? So again, Mr. Anup, can you please explain if you are around? How current COVID situation addressed Alzheimer's disease in elder people? Okay, so because of this pandemic, a lot of people with psychiatric disorders and many other disorders, cardiovascular disorders, everybody is affected because they are not getting timely attention, interventions. I assume that even people with, you know, very sadly, Alzheimer's disease would also, you know, be severely affected by lack of, you know, attention, lack of the necessary interventions in a timely way. Yeah. How does the fetus immune system respond to viral infection of mother? So it depends on whether the, you know, mother's immune system actually lets the virus to get to the fetus. But when I said maternal immune activation, even without the virus actually reaching the fetus, if there is high level of inflammation in the mother, in the, you know, the cytokines are known to reach the fetus and affect brain development in these you know, developing in this developing fetus and result in predisposition to psychiatric and neurological disorders.
Okay. Yeah.
And modulation or regulation of overexpressed inflammatory cytokines in NMOSD or RMS, will it help in managing disease? Of course, immunosuppression is one of the classical ways of maintaining these patients. Clinicians use these approaches, you know, every day, you know, using monoclonal antibodies or immunosuppressing steroids to manage these conditions, and they are pretty helpful. Yeah. How herd immunity for any patient's viral disease will be induced in a population. Yeah. So it's basically by getting exposed to the virus, probably low doses of viruses without actually becoming susceptible to the infection, just enough exposure to elicit a good immune response. By that, you have antibodies and cellular immune responses, which are ready to fight a subsequent infection. So that is how herd immunity will develop. Okay. As per the available literature, there are two major groups of SARS-CoV-2. Is there any variations on the immune response? Good question. There is no enough investigation into this area. Yeah, of course, a lot of investigations are going in, when are happening at this point, to see if these specific clades or strains of the virus result in different types of immune responses. But as of now, globally, and like discussed yesterday, it seems like there is no much variation as compared to susceptibility and immune response to the different strains. Is it true Indians are partially immune to COVID-19 due to BCG vaccine? Again, not enough evidence. Okay, so there has been speculation ever since this pandemic started that BCG vaccine could be protective. Not enough to support that yet. Because we are now seeing so many cases, all of us are vaccinated by BCG, right? Yep. Everybody in India is vaccinated by BCG. And in the last few weeks, you have seen a sudden surge in infections, even increase in mortality. So there's not enough evidence yet to show that. What is the role of co-inhibitory receptors in COVID? So can you please explain a bit? What do you mean by this? Guna Preeti, please. Are you around?
Hello, sir. Good morning, sir.
Yes,
I'm,
please.
I'm Dr. Chakravarti, working in Bangalore Medical College, sir.
Yes, please.
I work in screening center. So
Right.
I've been exposed to coronavirus since 4 months.
Okay.
And I've done RT-PCR 4 times.
Okay.
And all 4 times I got negative.
Okay.
And actually my wife and daughter were positive.
Okay.
I've been exposed constantly, but still I'm getting negative. So can you tell me the reason behind?
Where do you, so you work in the screening area, and so we have done a very good job of wearing the PPE properly and using it appropriately is what I feel.
Yes, sir.
And I should actually compliment you. And with respect to your mother and sorry, your wife and the child, do you think they would have got the infection from you?
No, sir. It is from my
Hello?
brother. We have a joint family.
Hello? Sorry?
Yeah, actually, my brother-in-law got positive,
Yes.
sir. From there,
Okay.
my wife and daughter got. No,
That's
my
right.
question is like,
Yes.
since I've been constantly exposed, but still my RT-PCR has
How, I know, I don't think so. I mean, yeah, that's what I should compliment you. You're not exposed. You have not exposed yourself to the infection. You have done a very good job of protecting yourself when you're doing your duty.
No,
So I
sir.
should,
Actually, I wanted to confirm you that
yes.
actually me and my daughter exposed to my brother. Actually, not from him. From my brother,
Right.
we
Right.
got an infection,
Yeah.
and even after exposing to us, he didn't take any distance from us.
Okay.
After exposing us also,
Yeah.
he did not develop any symptoms. So
That's
that's
wonderful. That's
what we
wonderful,
were.
isn't it?
What
Is
were
it?
yes, sir. But
Yeah.
the thing is, like what I thought was like might be he was infected earlier and he is asymptomatic. So because of that, even after exposing to us also, is
Uh-huh.
not developing.
Possible. Possible. That's what I was towards the end of my talk. There is, people believe there might. So, for one thing, is he has to look at antibodies, whether
Yeah,
he
yeah.
has antibodies, in his blood. Okay, that is one
Some
thing
of
you
them,
could do.
and another thing is, some of
Yeah.
them are telling his colleagues who got the positive and they're getting negative. They tested for this antibody test,
Uh,
but they did not find any antibody.
right. So it's possible that his body has never really got exposed to the virus, or
Actually,
or
he is exposed to my daughter, sir.
yeah, we have many such examples, madam. So so there are several amongst us who are exposed but do not show any signs of infection. So it's as I said, our innate immune system is very robust, right? If it is working well, so you won't even see a sign of what you looked at during the day, right? So it's possible. So be happy that his immune system is very robust.
Though he's been exposed continually constantly, but
Yes.
he's fine.
Yes, exactly. I mean,
Exactly.
there are many of us amongst us, right? So, yeah.
Yep. Yep.
Thank you.
Yeah. No.
Yeah. Yes.
So constant antibody-mediated response is enough to clear the virus. Not enough evidence yet. So, in fact, people are speculating that T cells may be more important. Because there is evidence that antibody response goes down after a few weeks. People, some people who have been exposed to the virus, so the antibody levels are going down, but and they see that T cell responses are sustained. So there's no clear evidence yet whether antibody only is enough to clear the virus, but it is important. Yeah. Constant wearing face masks, which might create hypoxia, will it affect? Very possible, right? When if you're not taking in enough oxygen, yeah, we know what all. So yeah, we should make sure that even though we are wearing these masks, especially the frontline workers who have to wear it for hours together, it is possible. So it's important that we take care of our health, take breaks, even if it is very difficult, breathe in well, and go back to our duties. So, yeah. In CD8 T cells, as it's shown, what is this guna? Yeah, I'm not able to understand that. Can anybody clarify that for me? What may be reason for higher asymptomatic cases and CFR in India as compared to other countries? Not really, right? I mean, this was expected. Now, asymptomatic cases are, the percent of asymptomatic cases are pretty similar across the world, most countries, which have seen high numbers of COVID-19 infected people. So we are not very different.
Okay. Yeah. Next.
What are FDA and EMA in vaccine development? If EMA probably is a European medical. I'm not very sure. I won't speculate. FDA, yeah, it's a Food and Drug Administration of the US, which approves vaccines, drugs, for human administration. There are strict protocols followed before approval. Yeah. Thank you, sir, for this informative.
Okay.
Which
What
is the
is
vital
the vital
force
force?
which will make the movement of lymph fluids
Fluid.
in our body? Who is this? Sorry.
Manohar Patar, like, yeah.
Yeah. So just like how blood flows under pressure is what I would speculate. I'm not very clear about that. Anybody in the audience can add. I'd like to learn.
Yeah, I would agree with what the statement you have made.
Yeah.
Along with the blood, it seems.
Yeah.
Yep.
Thank you.
Yep.
Thank you for. Okay, sorry.
Okay, I think
All right.
hopefully all the
Okay.
questions have been answered.
Yes. Yeah. I hope I've made justice to the questions.
Yep. Yeah, sir.
Right. Thank you so
So
much, Dr. Praveen. Yeah.
yeah, sir. So on behalf of the organizing team
Yeah.
and as a whole, like Devraj sir personally,
Yes.
so I would like to thank you
Right.
for such an elaborative presentation what you have made.
Yes.
It was really a very informative session that we could have it. So
Thank
once again, thank you, sir. Thanks a lot.
My
Thanks.
pleasure, and thanks for the opportunity.
Yes, sir. Thank you, sir. Thank you. Yep.
See you
So
all. Thank you.
yeah. Okay. And one maybe others, the other participant, maybe like probably will share you the link for tomorrow's session. Okay. And once again, I would like to thank one and all the participants who are here for being so robust. Though we started the session early morning, that in this winter climate, I would really appreciate the what the feedback from all the participants. Thank you, one and all. So, catch you up tomorrow. Thank you.